Okay, so hello everybody, and thank you very much for participating in the ENFIELD webinar, Bias in Medical AI. We are happy to have today some very distinguished speakers who will discuss about different issues related to bias in artificial intelligence. We encourage all the participants to enter into debates, to ask questions, to express their point of view, both after each presentation and also during the discussion session in the end of the webinar. Today's webinar will be moderated by Sabrina Bianchi from Maggioli and from me, I'm Martina Flora from Politehnica Bucharest. I shall be moderating the first part of the webinar until the break. We are going to have a short break of 10-15 minutes in the middle. And Sabrina is going to moderate the second session after the break. So thank you, Sabrina, for joining us and helping us moderate this webinar.
Sure, thanks Adina, and hello everyone.
Okay, now I'm going to ask Pankaj Pandey to give in the beginning a brief overview of the ENFIELD project. He is project manager for our project ENFIELD, and he is with the Norwegian University of Science and Technology in Gjøvik. So Pankaj, the floor is yours, please.
Thank you, Adina. Thank you for organizing this webinar and giving me this opportunity to present ENFIELD. I will share my slides to... Yep, I hope my presentation view is visible to you all. Yep, thank you. So once again, good morning all the participants. My name is Pankaj Pandey and I am a senior researcher at the Norwegian University of Science and Technology, campus Gjøvik. We have 3 campuses. And currently I'm managing this project called ENFIELD. ENFIELD stands for European Lighthouse to Manifest Trustworthy and Green AI. So basically the lighthouse is the keyword here, and which is the objective of the European Commission to bring together all the stakeholders from research, innovation and deployment in order to become a world reference in artificial intelligence. And if you look at the size of the consortium and the participants we have in this project, we have 30 partners from 18 countries participating in this project. And we have a nearly equal mix of industry and academic partners. And on this slide, you can see the various partners who are contributing to this project. Okay, to give you an idea of how the research and innovation activities are being carried out in this project is divided into 4 pillars. That is the 4 foundational elements of artificial intelligence. The very first one being adaptive AI. Second is green AI. Third is human-centric AI. And fourth is trustworthy AI. Likewise, the application of these foundational or you can say traits of artificial intelligence to specific industry domains is called as verticals or industrial verticals in this project. And there are 4 verticals which we are working on. First, it's healthcare. Second, it's energy vertical or industry sector. Third is manufacturing. And fourth is space. Now, in order to ensure synergy between all these 4 pillars and verticals, or you can say foundational areas of research versus the industrial domain, this is a pictorial depiction of how these different pillars and verticals interact among themselves. So the 4 pillars are the heart of this project. And then in the outer layer, they interact and contribute to the 4 verticals, that is energy, space, manufacturing, and healthcare. The key contributions or results which we expect out of this project are contribution to standards and certification, collaboration, networking and exchange with other projects, sister projects, development of a safety and security risk assessment framework, educational and training strategies, and of course, multiple novel AI algorithms or softwares, data sets, and so on. This project also has two third-party funding schemes. One is called third-party exchange scheme, which is for mobility of researchers, right up to the level of, you can say, associate professors. And total budget allocated for this scheme is 1.1 million euro. And we intend to fund almost 76 researchers over a period for mobility of up to 6 months. So minimum mobility required is 3 months and maximum is 6 months. And each researcher participating in the or selected for this scheme will receive a grant of euro 2,400 per month. So this is on top of the fellowship or scholarship they are receiving for their existing, let's say, PhD research. For their PhD research or postdoc or whatever position they are currently in. Now, at the same time, like TES, the third-party exchange scheme for researchers, we have a second scheme called as third-party innovation scheme. And this scheme is to fund innovation projects, the small-scale independent projects. And the objective is to fund almost 18 projects with a total budget of 1.1 million euros allocated for this scheme. And each project selected under this scheme will receive a grant of maximum 60,000 euro. So we fund almost 75% of the total cost of the project. We have already concluded first TIS call. Now the second TIS call is currently open, and this is open up to 17th of June, 1700 hours Brussels time. The very first QR code you see here is about the challenges or the topics which we have advertised to receive applications for second TIS calls. The second, small scale innovation projects. And the second QR code you see on the slide is the application link. So if you click on the very first, if you scan the first QR code, you will get a PDF document which provides the details of the various challenges we have announced. And you can go through those challenges, and whichever is applicable to you or you are interested in, please feel free to apply under that challenge through the second link, through the link provided in the second QR code. And to interact with us or to follow us on the latest news or developments, you can follow us on X, erstwhile Twitter. And we are also available, we do have a website like any other European project. But this website is currently under, you can say, shifting from one domain to other. So this may take some time or may not provide all the information readily. But our social media platforms like X and LinkedIn are always relevant, and you can follow us there, or you can keep an eye on the posts we make to be aware of the latest announcements. And so keep in mind the deadline for our second open TIS is 17th of June, 1700 hours Brussels time. Now, in addition to these two third-party funding schemes, that is TES and TIS, we do have several other events. For instance, we have hackathons planned under this project, 2 hackathons planned. First hackathon was organized by UPB in Romania last year. And the second event, the second hackathon, is being organized by Teltech, and it is in the next week in Estonia. Now, I have not provided the link to this second hackathon because the registration deadlines are over and the teams are already registered. But for your information, the second hackathon will be, I think, on 29th and 30th of May in Estonia, Teltech. Now, at the same time, we do have a couple of summer schools planned in the project. One is to be organized by BME in Hungary, and the second is by our partner TUC in Germany. I will come back to, in the next slide, I'll show you the information on the next summer school. And at the same time, our industrial partner Innova Plus, they have planned for several innovation workshops, which I think is not really open for the external participants, but is relevant for TIS and TES projects and the participants there. Now, the first summer school, which I just mentioned about, is planned from 28th of July to 1st of August at the Budapest University of Technology and Economics, Budapest, Hungary. And I believe you can still register for this summer school if you are interested in. And the registration or willingness is being accepted from students who are interested in participating in the summer school as students, obviously. And at the same time, we are willing to accept several expression of interest from potential speakers who would like to contribute to this summer school. And with this, I conclude the presentation, this brief presentation on introducing ENFIELD. If you have any questions, queries or comments, please feel free to post them in chat or follow us on the social media platforms I mentioned earlier. Thank you.
Thank you very much for your presentation. Very concise and extremely informative for all participants. In case you have any questions related to the project, because obviously there may be a lot of questions, I would invite you to put them in the chat. So we are going to keep questions, live questions, only related to presentation. Sorry. Now, the next speaker is Professor Bjørn Hofmann. He is a Norwegian professor in philosophy of medicine and bioethics with special interest for the relationship between epistemology and ethics. He is affiliated with the Center for Medical Ethics at the University of Oslo in Norway and the Department of Health Science at the Norwegian University of Science and Technology. We are delighted to have Professor Hofmann with us. Alex, please stop the recording. Yes, Pankaj, please.
Yes, thank you, Professor Hofmann, for the very interesting presentation. I noted some keywords from your presentation like automation bias and patients or doctors not being fully aware of the outputs they receive from, let's say, AI systems. So recently there has been a trend in using AI-based systems, particularly for radiological applications, radiology diagnosis and stuff, including in Norway and several other countries. So how do you see this in terms of the ethical or philosophical challenges? And second part of this question is, do you see the role of contestability in terms of addressing some of these challenges posed by this AI being as a radiologist for the doctors as well as for the patients? Thank you.
Excellent question. Thank you. I've been working with radiologists for some years, and initially they were extremely afraid of AI because they were afraid of losing their jobs. Now they have changed their attitude. They're very much looking up at it as a tool, but more than a tool, also an assistant. And maybe some of them are getting a bit too enthusiastic. I think there are lots of very good examples where it can be very helpful. For instance, reducing double reading of mammograms. You can reduce from 2 to 1 radiologist assisted by AI, for instance. I think it's very important to not follow the hype and say, because I've visited some conferences in radiology and they ask the vendors, does it have AI or not? But the question is not, does it have AI or not? But the question is, on what material is it trained? And is this material relevant for the patient group that I am going to encounter in the near future? So I think that's very important, and that's also why contestability is very important, to try to be very critical with regards to the characteristic of the AI system, and of course that can be hard to quantify or to verify. So in my view, any types of good verification is key in order to document the effectiveness, the safety, and the efficiency of and quality, of course, of AI systems. So that's very important. So thank you. That's a great question.
I have a follow-up question too, if you don't mind. So how do you see the implementation of contestability mechanisms under the ambit of laws like EU AI Act or GDPR regulations? Do you see these regulations or laws supporting this kind of mechanism or being on the other side of the fence?
I'm not aware of the details, so
Okay.
I think I should be careful answering that question.
Okay, thank you. Thank you. I do not have any more questions. Thank you very much.
Okay, I would have also a question, or maybe two, for Professor Hofmann. First of all, in one of your slides, you mentioned briefly something which, well, concerns me. I mean, I'm thinking about it from time to time. What if the people implementing the system or feeding the data or gathering the data are not aware of the biases that they have? So this would be the first question. And I'm going to have another question. So what can you say about that?
No, that's a problem. You don't know what you don't know. That's the ignorance challenge. And of course, I think a lot of the work that has been done to measure and identify biases can at least help somewhat with that. The problem is for those who are not aware and use this. And as I indicated, some of these tools are quite fragmented, so they may be able to identify specific types of biases. For instance, gender biases is maybe easy to identify, but other types of biases related to socioeconomic factors, for instance, may be harder to identify and may be ignored. So that was why I think we have to pay special attention to the ethical issues I mentioned at my last slide.
Okay, thank you. And the second question, in general, in the literature on the resources existing on the web, there are many proposals on how to classify bias, both done by researchers, by companies, by very reputable organizations as the one you presented in your slides. And I search in order to try to find an ontology for bias, because as you know, there are ontologies in all sorts of domains, especially related to AI, but I failed to find it. I mean, is it true that there is no ontology, and do you think that it would be a good step towards a kind of attempt to regulate the different kind of biases?
Yes, I think this is a very good point. With all emerging fields like AI, at the beginning there is no structure for basic concepts, and ontologies are floating. After a while, they will, in a way, emerge. But right now there's a jungle out there with ways to define and ways to identify and ways to try to mitigate. I think there will be some kind of standardization after a while, but efforts to try to standardize will be very helpful, especially early in the field. We've seen that also. I mean, the COVID pandemic, there was a lot of uncertainty. It was called by many names, and then we came down to SARS-CoV-19. So this will happen also in this field, and the sooner the better. And the more hands-on the professionals who work with this are, the better it is. So far, the psychologists have been most elaborate on classifying biases. So if you look at the literature, a lot is imported from psychology. Probably this will change when the AI community themselves are able to look at and classify biases. So I really very much encourage all the work on establishing ontologies.
Thank you very much for your answers. Are there any questions? Thank you very much, Professor Hofmann, for your presentation and for your answers. Now we shall go to our next speaker. Our next speaker is Professor Sören Möller. He holds a PhD in mathematics. He's a biostatistician and associate professor with the epidemiology, biostatistics and biodemography, Department of Public Health, University of Southern Denmark. So, Professor Möller, the floor is yours.
Thank you. I hope you can hear me. Yes. Then I will share my slides. One moment.
Yes.
Yes.
Yes, we can see it. Thank you.
Thank you. Yeah, thank you a lot for the invitation, and sorry for the very long name of my talk. I could have instead called it that be aware of the aim of your prediction model in health when you interpret the risk of biases. Because the idea is that, as you all know, we often like to predict outcomes in healthcare, and it's becoming even more prevalent due to different kinds of AI models and due to larger and more comprehensive data sets and computational developments making frequent and on-demand prediction feasible, and because everyone, or at least many people, like predictions. Since both health professionals, politicians, and other patients seem to like predictions in many cases. And the thing I want to point out in this talk is that it carries some risk for biases because different prediction models have a different aim, and the aim has influence on the precision we can expect from a model and the reliability of the model and the difference in balance between risks and gains of a prediction model. So it's important to be aware of what's the aim of the specific prediction model, to which in most settings related to this group at least will be some kind of AI model, and then also hold fast in that aim so that you not change the aim afterwards. So evaluating a model for one aim and then applying it in practice for another aim could give problems. And for this discussion, I have split the aims a prediction model in health can have into 4 possible aims. It is not a clear distinction you could lie between, and maybe there are other aims, but those are the ones I'm mostly seeing when working with clinicians and clinical epidemiologists, that sometimes you just want to model the population to predict on a population level. That's very classical in epidemiology. Things like extending time trends in diabetes incidence, taking into account demographic changes, very classical type of questions. Sometimes then also we want to model the individual but predict on a population level. So we want to, in our modeling strategy, utilize very detailed information on individuals, but the aim is still to say something on a population or group level, thinking like predicting the expected characteristics of future cancer diagnosis in an area for hospital facility planning, for instance. So it's not that we want to say something about a specific patient, but still we want to use very detailed information. But the part where AI is becoming more and more relevant is doing something on the individual level, where you can model the individual to decide individual diagnosis from a prediction, things like predicting osteoporotic fracture risks to refer patients to further investigations. We have had quite a number of projects here in Denmark with that aim. Well, then the result of the prediction model is to decide on extra scans and closer investigation, but there are also in the fourth group models where we model the individual to decide individual intervention, so things like predicting the effect a drug will have on a patient, to guide the choice of chemotherapy in a cancer treatment. So where the prediction model directly influences a treatment decision and not just is one step in a diagnostic process. And those 4 different aims have quite different behaviors, and the risks for biases and the drawbacks for the patients increase if you go down on this list of prediction models. So if we are in the very classical case predicting from a population to a population, then that's very classical and often done by regression models or time series approaches, but it also could meaningfully be done by AI, and maybe we could get a better prediction model on the population level, which would be quite nice. But basically we have been good in this prediction for decades. We can get better but it's not doing this forecasting expected incidences or prevalences in diseases or general population forecasts in demography, fertility, and mortality forecasts. That's something we are quite good at, and we have been quite good at it for at least some decades. So here the risks are mainly that there are unexpected changes, shocks, and time trends. So it's not so much a risk of the model, it's just that most of those models expect things to behave like the data we train them on. So for instance, since its relation to COVID, there of course have been a lot of predictions that went wrong because we forecasted expected number of infectious disease patients, and then suddenly we had many more, and of course the forecast couldn't plan that. So the risk here is mainly that if you believe too much in those predictions, then you can get a bias that assumes that everything is as it's normally. So you can overlook the risk of being resilient to shocks. And also you could extrapolate to non-representative populations that either could be extrapolating outwards, developing a prediction model on some populations and then trying to use it in much bigger areas, or the other way around, that it doesn't hold for some subgroups, that you, for instance, predict trends of incidence of some disease, but have a growing subgroup in the population that maybe has a different genetic background and very different risk, and then you wouldn't be aware that there's a different behavior. So those risks are mainly epidemiological risks and not so much risk in the AI models themselves. But if you then go to modeling the individual, to say something on a population level, then we still are in a quite similar setting. Basically, we are just using more information. We're using detailed individual data, but the questions we want to answer are still very similar. To say something about a population trend, often, for instance, in more detailed planning of healthcare resources, because what we are interested in is not only how many patients will there be, but also how many of them will require advanced treatment, things like that. So getting more detailed information out by using that in many cases, what we could use as input for those forecastings on a population level could be very detailed individual data. One of those places where it's done this way is, for instance, predicting changes in population in Denmark, where you, in some cases, use quite detailed fertility forecasts on very detailed subgroups. So trying to model the very detailed subgroups on the expected fertility and then combining that on a population level to get population forecasts. And here the risks are mainly that you then decide on which parts of the population have characteristics that are important enough that it makes sense to extrapolate them to the full population. Then, so you might, for instance, if you do fertility forecast taking into account some characteristica, if you choose the wrong characteristica, you might be worse than if you just had done a population level model, because if you imagine that education has a high influence on fertility and very focused, build your model to take into account changes in education, then, but that's not the driving factor in fertility development in your population, then it could behave worse than a population model. And also extrapolation to the future may overlook population changes. This utilizing detailed information also means that your models very often will be closer related to the population composition. Then, so I think that in the first type of models, this mainly would be a shock for the thing you want to predict. Here also large changes in some of your predictors could change things. So you have to be even more careful when extrapolating that you don't overlook some systematic changes or some shocks. Where it becomes more interesting and more typical using AI models is if you want to model the individual to decide individual diagnosis. And that's the part I have been mostly involved with, because there are a lot of ongoing prediction models in Denmark where we try to use a lot of health information on the individual to predict the risk of a disease with the aim of, for instance, doing a kind of two-step screening process. That's predicting you are in a high-risk group, and because of that we invite you to our screening. Or you are in a high-risk group, so because of that we should invite you to 2 visits at the hospital for checking up on your diabetes each year instead of only 1 visit. Things like that. So it's not influencing the treatment, but it's influencing how much focus there's on a screening or diagnostic setup. And that's, of course, has been possible for a long time, but it's starting, at least here in Denmark, is starting to get more and more applied in practical clinical work. And the risk here mainly that there could be a systematic bad performance in subgroups, and of course that could get some inequalities and result in some biases, because then there might be subgroups where the model expects them to be of very low risk, and because of that you don't invite them to a screening, but biologically they're at quite high risk, but just due to some factors that weren't observed or weren't available when you trained your model, and then you will make things worse for them because you will offer them less screenings than they maybe would have been offered in general, because they are not anymore considered as high risk. So if you, in many cases, if there's part of the population you classify as being high risk, there are also other parts you classify to be low risk, although if you wouldn't have done these things, most of the population would be considered average risk persons. In this case, it's false positives mostly will be benign because the next step often is a not very invasive diagnostic procedure, but it also includes some research use if you have a lot of false positive predictions. It depends quite a bit on the next step, because there have been some cases where the next diagnostic step is a very invasive step, having some drawbacks. So it's not always this case, but in most cases the next step will not be very dangerous, because it often is some scan or an extra talk to a healthcare professional or something like that. False negatives, of course, are undesirable, but in most cases they will be undesirable but similar to the program not assisting. So the patients won't be worse off being a false negative than if there was no prediction model at all. That's again not always the case, but that's often the case, and means that false negatives maybe aren't so bad, at least on a population level. If you then go to the last step and use the model to decide on interventional healthcare intervention, then that's not very common yet, but there are a lot of wishes to do it, and there are some places where it's done already. There are places where you use some of the same osteoporosis high-risk identification models we use here for screening, but where the next step isn't screening, but is starting up pharmacological treatment. Maybe because screening facilities aren't really available, so if you're considered high risk, the next step is we give you medicine. There are some places where that is done, or at least has been done. Or predicting the effect treatment, for instance, a chemotherapy treatment will have on a cancer, so to decide which one to choose. And here the risks become a bit more dangerous, because you could mispredict atypical patients in both directions, because if you mispredict atypical patients and then withhold their treatment because you don't think it will work, or you mispredict atypical patient and then you start up a lot of treatments with side effects, although they didn't have the disease. In both cases it can be quite dangerous, because now it's on the individual quite important that your model works on each individual, because if your model works on 99% of the individual but is does very bad things for the last percent, then that would be a problem, because then you would do dangerous things to 1% of your population. So in this case, both false positives and false negatives can be highly problematic, because initiating unnecessary treatment can be quite dangerous, and withholding necessary treatment also is quite dangerous. So in this case, the balance typically is much stricter than if it's just for a diagnostic process. In general, my suggestion is that one of the questions you should ask when doing a prediction model in health and considering if it makes sense to implement it and evaluate, asking the counterfactual question on the model's existence, then how many and which patients would be better or worse off if the model did not exist? Because that can be a very helpful question in a discussion, then to decide if it's a good idea and what to be aware of if you want to implement it. Mathematically, that's quite easy. It's false positive and false negative rates, but the weight, utility, and loss functions can be very different between false positives and false negatives, and they can be quite hard to quantify. It's very hard to say for this use case, false positive is 10 times as bad as a false negative. That information can be hard to get out of practitioners. So in many cases, maybe this is more a discussion point. Also because the behavior can be very different between different individuals or subgroups. And often it's not entirely clear what you want to compare with. You can compare with guessing. That's basically if you very classically report an receiver operating curve and an AUC value for a model, then you compare to guessing, and the worst case not very realistic. In very few cases, if an AI model wouldn't exist, what you would do instead would be to guess or to flip a coin. So maybe that's not the most informative comparison. On the other hand, you could compare with an oracle. That's the best case. That's not at all realistic. If we would be perfectly good at making the right decision without a prediction model, then probably we wouldn't build a prediction model. So in most cases, possibly one should consider the counterfactual situation. If we implemented our AI model instead of the current practice, how many and which patients would be better or worse off if we wouldn't have done that? That can be very informative, both to see the general gain, but also identify subgroups where our model might have biases that are so bad that they make things worse for some individuals. Or if there are other suggested approaches, if you would have to decide between two AI models or decide between an AI model and a classical screening program, then considering if we did this or this, which patients would be better or worse off? And especially those that are worse off, that should be hopefully quite few, and one should be aware of them, because that's the people, the biases of our AI model will do bad effects for. So in general, my conclusions are that prediction models have very different aims, and that's fine, it makes sense, but those aims strongly influence the risks involved and what to be aware of and which biases are worse and more important or less important. So be very aware of the aim of your model and communicate it clearly also to the practitioners. If you build a model that's thought on a population level, be clear to communicate it so that not some very predictions loving politicians then end up implementing it at the individual level. That's one of the problems, not in healthcare, but currently in Denmark there's been problems with government trying to estimate the worth of houses from some models that were quite good if you want to know how much are all the houses in the city worth, but when you try to then implement it for taxation on each individual house, it goes very wrong for quite a number of houses. And the same thing could happen in healthcare. So be aware that if a model has one aim and it's good for that aim, that doesn't mean that you can use it further down the chain necessarily. So reevaluate your risk and the utility of the model when you decide to extend the aim or consider extending the aim. Yeah, that's my conclusions. Thank you.
Thank you very much for your presentation. Question, please. Yes, Bjørn?
Thank you so much. This was really interesting. Just a question. You mentioned DEXA, right?
Yes.
And of course, for many of the data sets, there's a problem with overdiagnosis. And this is not measurable on an individual level. So it goes back to Adina's question also with regards to what about the unknown unknowns? We know it's there on a population level, but we train the algorithms on that, and it's very hard to compensate. So any type of advice would be very welcome.
Yeah, it's a clear problem, and I think one of the it's an unknown, so it's quite hard to do, but at least one can use some triangulation approaches to try to detect which subgroups there might be our diagnosing, and then maybe use that in calibrating the models. So for instance, we have had some studies where a prediction model was used to offer screening to people in the population, and then of course some of them said yes to the screening and were DEXA scanned, and then maybe our diagnosed and our treated, but there also are groups that ignored the latter because they didn't want to be screened. Then so such groups can give at least a type of natural experiment, because if the whole thing works, then you would expect there to be a outcome benefit in those that got offered the treatment, and you also, in many cases, from a clinical or biological point, have an expectation how big that benefit should be, and then you will see the benefit is much smaller because the process gives them our diagnostic and our treatment. But then that can give an information on how much is that our treatment effect, and if your data are big enough, that also would give an information at least on core subgroups of the population. Is this our treatment larger in women than men? Is an age difference? Or what we saw in our concrete study, for instance, was that there was a geographic difference, that in larger cities more people said yes to the screening, resulting in a higher treatment effect, but probably also in more our diagnosing. In the countryside, more people said no to the screening, avoiding our diagnosing, but also avoiding gain from having the program at all. So that kind of things can at least partly inform this problem. But I agree that that's a big problem, because we only can observe what happens in reality. We can't really do counterfactual observations.
Thank you. Other questions? Thank you very much, Professor Möller, for your very interesting presentation. Now we have to move on. The next presenter, Sofia da Rocha, unfortunately she cannot attend the webinar because of some unforeseen event. So I would invite the next speaker in our webinar. So Barbara Draghi, she is from Brunel University, London, and Medicine and Healthcare Products Regulatory Agency in the United Kingdom. She's a data scientist and machine learning engineer who is passionate about data-driven healthcare and special interest in bias and fairness within AI system, machine learning, and synthetic data. So, Barbara, the floor is yours.
Thank you, Adina. Thank you so much for the introduction. I will now try to share my screen. Hopefully it's gonna work. Can you all see my screen? Great. So, good morning, everyone, and yeah, thank you for inviting me and having me today at this second ENFIELD webinar on bias in medical AI. As Adina said, I'm Barbara, and I wear two hats in my professional life. So I'm a data scientist at CPRD, which is an MHRA-sponsored service that provides real-world data for public health and clinical studies. And in parallel, I'm a part-time PhD student at Brunel University, London, where I research synthetic data generation methods to tackle bias in patient data. So today, I want to give you an overview of how synthetic data can help us to mitigate bias in primary care records, improving the fairness and reliability of medical AI. So in the next 15 minutes, I will walk you through CPRD, explain why synthetic data is so important for medical research, and then we will explore the problem of bias in real-world patient data and show how synthetic data can help somehow to mitigate this bias. And finally, I want to give a brief introduction to Emanuele, one of the last CPRD synthetic data self-service products. So let's begin with an overview of CPRD and the role of synthetic data. CPRD stands for Clinical Practice Research Data Link, and it is a UK government research service supporting both retrospective and prospective public health and clinical studies by academics, industry, and regulators worldwide. It is sponsored by the Medicines and Healthcare Products Regulatory Agency, which is the regulatory body of medical devices in the UK. CPRD is one of the largest sources of primary care data for public health and research, and they have been collecting electronic health records from GP practices across the UK for more than 30 years, which covers roughly the 25% of the UK population. And this incredible amount of data is an unprecedented opportunity for innovation to improve patient care, discover new treatments, understand disease progressions, and so much more. But as we all know, healthcare data is not easy to access, and the Data Protection Act in 2018, which is the UK's implementation of the GDPR, General Data Protection Regulation, strictly controls how personal information such as CPRD data is shared across research communities. So data holders cannot directly share their data with the machine learning community due to detailed policies to maintain the privacy of individuals, of course, and these make it tough to build and test AI models. And synthetic data offer a way around this to some extent, because synthetic data mimics the characteristics of real data without containing a one-to-one mapping with real individuals. And that's why in 2018, CPRD started experimenting with generative model approaches on our primary care data, and our very first proof of concept showed that we could capture key patterns in diagnosis, prescription, and demographics, and by 2020, we published the first 4 pilot datasets to medium fidelity samples and 2 high fidelity synthetic datasets focused on COVID-19 and cardiovascular disease risk factors. Of course, more details about this data are available on our website at cprd.com synthetic data. But let's just say that now at least we have a rough idea of why synthetic data is important, and especially when it comes to privacy and data access. So I guess now we can, shift gears to the other side of the coin, which is bias in real-world data. And, well, we are all here today because we all agree that bias in AI matters. It is a problem, and I'm sure each of us could list examples off the top of our heads. Still, it's worth, a quick reminder that over the past decade, we have seen biased AI systems show up everywhere. So first came the Compass system, one of the criminal risk tools which consistently labeled Black defendants as higher risk of reoffending than their white peers, even when the underlying behavior was the same. Then we saw high-risk algorithms that learned from years of resume data and quietly downgraded women's applications because fewer women had applied in the first place. And after that, recent COVID pandemic happens, and medical devices like pulse oximeters turned out to give less accurate readings on darker skin tones, literally putting these patients at greater health risk. And I would say that what ties all these failures together is simple. The models were trained on historically biased data, and when your inputs are skewed, your prediction will be too. So if we carry this problem into a synthetic data generation pipeline, let's say if the data that you feed into your generator are already biased, there is a real risk that you will simply reproduce or even amplify this bias in your synthetic records. And if you train a clinical model on biased synthetic data, you will end up with unfair outcomes for the biased groups. In healthcare, the stakes are high. Biased data can lead, for example, to wrong recommendation systems, steering clinicians down the wrong path, or misflagged at-risk patients, either missing those who need help or over-alerting those who don't, or to ineffective treatments because we are testing therapies on the wrong subgroups. And that is why it is not enough to generate synthetic data, but we need methods that detect and correct bias to be integrated in our pipeline. And given this risk, the next question is, how can we use synthetic data not only to reflect real-world patterns, but to identify and fix bias? So let's see how synthetic data methods can help in that. So I would say that the power of our synthetic data pipeline is that it's modular, meaning that you can plug in steps or swap components to address different needs. And in a very simple way, I want to give you an overview in how we can turn, a standard synthetic data pipeline into a bias-aware one. And well, we start by measuring, the bias in the real-world data, examining disparities across demographics, conditions, or any other feature of interest. And later we're gonna try to see in a more detailed way how we can do that. And we translate these bias measurements into formal constraints, and which our generator is extended to accept these conditions and produce synthetic samples that kind of satisfy them. And of course, we need a post-generation evaluation because we need to evaluate the synthetic data bias and fairness using ad hoc metrics. And with these additions, our pipeline can evolve from a basic synthetic data flow into a fully bias-aware framework, trying to detect and mitigate bias and verify fairness at every stage. And now we can have a closer look at the method that we are investigating to detect the bias. So I've been researching two, let's say, complementary methods for measuring bias in real-world source data. One is based on uncertainty analysis, and the second one is more focused on representation bias. Why two different methodologies? I would say that when you have large data set, let's say tens of thousands of records, uncertainty analysis is the go-to approach, because you can split the data into a training set and a holdout validation set, train a probabilistic model on the training portion, and then measure prediction uncertainty on the holdout set. And records with high uncertainty, indicated by wide confidence intervals or divergent predictions, point us to the exact patient profiles. For example, can be like middle-aged men with unusual symptoms combination, where bias or data sparsity has the greatest impact. And uncertainty analysis can work, but if you have large data enough to extract an independent validation set, which is why we probably need also something more which can work on smaller data. And in smaller data, when an independent validation set cannot be extracted, we can use something called representation bias analysis. In this case, what you do is you can select sensitive attributes, one or even more, such as gender, ethnicity, and also outcome label, for example, stroke or versus no stroke. And then you can calculate some metrics, for example, coverage metrics, by comparing each subgroup's share of the sample against its expected share in the true population or any other reference population. And for instance, we might discover that women over 65 make up just 5% of our records, but should represent 15% according to the national demographics. And despite the differences of these two approaches, I would say those methods are very complementary because you can use one or the other, and both will lead to the same sort of deliverable, like a clear list of quantitative conditions that are going to say we need more female patients under 50 by 30%, or reduce the overrepresentation of urban residents in high-risk cohort, for example. And once we have generated these bias quantification conditions, we feed them directly into the generator to synthesize new synthetic data corrected for the detected bias. And after generating synthetic data, we can move to the evaluation part. So in terms of evaluation, I would say it's really tricky to, as Professor Hofmann mentioned before, there are many metrics, that are available to measure bias, to measure fairness, and it's really important to understand, according to me, what is that you want to measure. So I spend so much time reading about bias and fairness, also to understand the difference between those two concepts and how you can measure bias. For example, starting on the data, you can measure the representation bias on the data. But then when it comes to measure fairness, of course, you need different metrics, because fairness is something that you measure on outcome of predictive models that are trained on your data. And so for the evaluation part, I developed this pipeline where we quantify representation bias for some synthetic data, and then we train predictive model on the synthetic data and quantify fairness on the outcomes. So, for example, there are many metrics available, as mentioned before, and you need to choose the right one for your scenarios, which could be, for example, accuracy parity, predictive rate parity, equalized odds, but you need to be very careful in choosing the right metrics for your problem. And with these slides, I just want to propose some very basic results, just to give you a flavor of, how this framework can work in a case study. So, for example, these are the results that I collected by the application of the described framework to CPRD cardiovascular disease risk factor dataset, which is a dataset focusing on, I said, cardiovascular disease risk factor. And the bar chart here shows the results obtained by testing our framework in selecting ethnicity as the protected attribute, like the sensitive attribute to investigate the bias for, and stroke heart attacks as the target disease. So the green bars here represent the spread distribution of ethnicity in the original data, whilst yellow bars represent uncertainty analysis outcomes. And the idea is that if the yellow bar is higher than the green bar, we need more cases of these groups. And of course, if the yellow bar is lower than the green bar, it means that there are enough data for them already. Blue bars represent the distribution of ethnicity in the dataset resulting from the application of bias-aware framework. Therefore, they represent the dataset corrected for bias. And comparing the blue bars to the green one shows that our approach is working. So just as an example, if you look at, the Indian group here, the green bar represents the original data, and the yellow bar tells us that we need more subjects belonging to this category, and blue bars increase the number of subjects. Our approach, of course, aims not only to generate synthetic data where bias has been reduced, but also to obtain better predictive models for desired disease. And that's why in the evaluation pipeline, we also introduce some performance and fairness assessment. And here on the left, we have a classification accuracy result showing the results obtained by comparing the original data and the data corrected for bias in predicting stroke. And as we can see, our approach increases classification accuracy while maintaining reasonable confidence intervals. And on the right, I just provided some results on fairness matrix, showing that, for example, if you're looking at the equalized odds for 9 different ethnic groups here, in the original data chart, there is a widespread. Some groups fall well below the ideal parity line at 1, meaning that the model's true and false positive rates differ significantly across the population. And in the corrected data chart, every group kind of clustered tightly around the parity line, showing that those disparities have been addressed. As I said, this set of results was just to give you an idea of what I was talking about before, but additional results are documented in the paper that I cited here, if you're interested. And more findings are also on the way, because we are, as I said, this is a research work, and we're still working on it, and we have worked on different settings, different clinical scenarios. More findings will be published soon, so in case you're interested, just stay tuned. And as I said, so this is a research, so it's not yet implemented in CPRD synthetic data service, but it will be soon in the future. And just before we conclude, I'd like to spend a few words on Emulate, which is our self-service synthetic data platform. So right now, Emulate offers a no-code web-based interface powered by our synthetic data generator, so where you can upload your tabular data and generate high-fidelity synthetic record. And if you'd like to try the prototype, you can just email inquiries@cpre.com with Emulate EOI in the subject line. And in the future, as I said, bias mitigation strategies will be integrated. And well, I would say thank you all for your time and attention. Again, it's really a pleasure for me to be here. And I hope you found this interesting, and I appreciate you sticking with me. So now I'd love to hear any question or feedback. And well, yeah, thank you again.
Thank you very much, Barbara, for your very interesting presentation. Question, please. Yes, Bjørn.
Thank you, Barbara. This was really excellent and very nice overview. I have more or less the same question as for Sören with regards to some of the blind spots which are not observable on an individual level. For instance, overdiagnosis, representing or introducing a bias which is very hard to get rid of. So that's the first question. But the second question, I mean, in your case, accuracy increased at the same time as fairness increased, but for a lot of cases where you make synthetic data, you have to have a trade-off. And if you have any reflections on how to trade off, if you have to reduce accuracy in order to increase fairness, what then to do? What would your advice be?
Thank you, Professor Hofmann. I guess, I mean, that's a very, very good question. So I will start answering your second question first. Well, I guess that's one of the main compromise that you have to make when talking about, generate synthetic data. I think it's really important to define, let's say, what is your final goal? So what do you want to use your data for? What is your use case? Do you want, your data to lead you to have a fair model, or do you want to have high accuracy? And you have to make a choice, according to me, before synthesize your data in a way, because as you said, it's a trade-off. And when you have data, data cannot be used for everything. So it really depends on the specific use case, if that makes sense. And coming to your first question, can you please remind me? I don't think I got it correctly.
No, because one of your initial points was identifying the biases, and of course you want to do so on individual levels if you can. And you used the example of gender, for instance, or race. But with regards to overdiagnosis, i.e., cases which have been identified as diseased and treated as so, but didn't benefit from the treatment at all because they would have died with the condition and not from the condition. And these cannot be identified on the individual level, only on the populational level. And this represents a bias in the data set which you can't get rid of, and it will follow you, and it will follow you also when you generate synthetic data. So any reflections on that will be most welcome, I think. It's not an easy question, I know, but you may have thought of it.
No, that's very, very interesting. That's a very interesting point, actually. And of course, we started focusing on individual level because, of course, we're dealing with, patient data at the patient level. For us, it was the first, point to address was, trying to detect those type of bias and address that type of bias. But of course, what you're talking about is very important, and it's definitely something that we are aiming to investigate soon in our research.
Other questions? I would have two questions. The first of all is, in fact, an acknowledgement more and the question about the platform Emulate that you have mentioned. I didn't know about it, so it's quite interesting. So if I understand correctly, it can generate some synthetic data for tabular data. Is this correct?
Yeah, that is correct. Yeah.
Okay. Any kind of tabular data?
Yeah, actually, we developed this platform having in mind any type of tabular data. So yeah.
Okay. Thank you. And the second question is, I mean, your experiments with introducing all these metrics for measuring bias and so on, they were done for some specific disease, if I understood correctly. Stroke, you said. Is this correct?
Yeah, so I would say depending on the data set, of course, when you define the use case, so stroke was investigated, for example, in the case of the cardiovascular disease risk factor data, because a stroke is the outcome in that particular data set. In the case of COVID-19, the outcome was a COVID-19 diagnosis. In case of other data set, you just define the target label in your data.
Yes, yes. My question is, do you think that your approach, your proposed method, is independent of this particular disease, or it would have some kind of dependencies and you would need some kind of fine-tune for the specific use case? I don't know, stroke, multiple sclerosis, or I don't know, heart attack or something like this.
Oh, yes. Okay. Yeah, no, thanks for the question. Very interesting. I would say it depends on the approach that you choose. So in my framework, for example, I investigated two different ways of quantifying the bias, right? The first one, uncertainty analysis, is dependent on the target that you choose, because in a way you are investigating the bias that is affecting the prediction of that particular outcome. Can be done for also multiple outcomes, though. It can be like more than one. In the other case, if we just focus on the representation bias, the approach is independent from the target label. So the target label in that case is just used, to quantify at the end the fairness, because to quantify fairness, as we know, we need, to identify a particular target to predict. If that makes sense.
Yes, yes, it's quite clear. Thank you very much. Other questions? Okay, if there are no other questions, then what I'm proposing to do is to move to the next presenters and have our break afterwards. So we have a presentation. Thank you very much, Barbara, for your presentation. We have now the presentation of Konstantina Remoundou. She is a statistical researcher at the Institute of Communication and Computer Science, Greece, and Emanuele Koumantakis from the Department of Clinical and Biology Science, University of Turin, Italy, and he is also a PhD student in experimental medicine and therapy. Now, the floor is yours. Please.
Thank you, Adina, and hello everyone from Greece. So let me share my screen one moment. I hope you can hear me all well.
Hear and see you very good.
Oh. Okay, thank you. And I hope you can see my screen. So today, along with Emanuele, we are going to present you the most common biases in electronic health record databases that we have actually identified ourselves through a case we are working on prediction in readmission cases in ICU. One moment. Okay, so this case was an idea coming from the healthcare vertical from ENFIELD through the exchange scheme. We had the opportunity to work with Emanuele on the prediction models regarding ICU readmission cases. We have collected studies implementing models, deep learning models for adults. So what you are going to see is mostly results from that research. I'm not going to stay here for very long because this is an ongoing still research we are doing. So using, we have identified cases in which, and other reviews, in which they have also seen that the risk of bias is very high in most of cases when developing a model through AI. As you can see here, the study from Kumar identified that 50% of their studies, their publication and so on, have identified a high risk of bias, while only 20% had a low risk of bias and a weak algorithmic design. Also, this is a case as well for the publication for the review from Chen et al., which they have studied 555 published neuroimaging-based AI models, and they have identified that 83% of them has a high risk of bias, with very few included only external validation. And lastly, because I don't want to be very long with that, in the systematic review from Chen F. et al. In 2024, through 20 selected studies, they have found that only 8 of the studies applied performance metrics in order to see those biases and see what was wrong. But the rest of the studies, the 12 rest of the studies, has made only a test for some form of statistical parity. So these findings actually show how much important it is to be aware of the healthcare bias from AI, in order to fight mitigation and have a clear mind when doing a research or developing an AI model to see and try to be fairness within the data and for the patients, to have a fairness for the patients. So in order to present that, we will present you a dual approach actually. Firstly, for the clinical medical approach, I mean what the medical and staff and the research staff should have in mind in order to be more correct against the data. And the second approach will be the statistical and more model development approach. So I'll give the floor to Emanuele to show you the medical approach.
Thank you, Konstantina, and good morning, everyone. Can you hear me? Okay. So for the medical approach, we wanted to give like the point of view of clinicians when it comes to apply deep learning models or in general AI models in their clinical practice. So should they trust the results from previous studies that developed those models? So can they expect that that results are reproduced in their hospital, in their setting? And so this is maybe the main issue, the first issue that comes to the eye of medical staff. So we can define 3 criteria for reproducibility that are to achieve in order to have a full reproducibility of the model, that are the technical, statistical, and conceptual reproducibility. Regarding the technical reproducibility, this is defined as the ability of another independent research team to reproduce the same results using the same model based on the documentation shared by the developer team. So in order to have full technical reproducibility, mainly 2 things have to be shared. The data set, so it should be a publicly available dataset, and the code of both data preprocessing and of the model, of course. There are some issues, even if they are, for example, if the dataset is publicly available, and this occurs few times, because the dataset goes through their identification processes in order to be publicly available, and this could limit their usefulness. For example, in our case of intensive care unit readmission, we should be able to understand if different admission to ICU are from the same patients in order to label the positive or negative outcomes. So if after the identification of the dataset we cannot do that, the dataset cannot be useful for that task. Another thing that I'm thinking about is when numerical continuous variables are treated as categorical after the identification, so they can use their meaning. Regarding the code, even if it is shared, it may have some bugs and cannot run correctly in some devices, in some platforms. So if we can see what we retrieve in our systematic review, almost all of studies had publicly available datasets, and almost, if we can switch in the next slide, we have all the studies that theoretically have publicly available datasets. I say theoretically because for one example, in one study, we have to receive the approval of an ethical committee before retrieving the dataset, so it could be more complicated than in other cases. But we can see that only 4 studies of the 24 that you included were published, also shared the code on GitHub. That's the main platform. So it makes it impossible to have the full technical reproducibility. Regarding statistical reproducibility, this is the ability to obtain statistically equivalent results under resampled conditions, and this is obtained after internal validation. This is a topic generally addressed by studies, so also in our review almost all of the studies performed the internal validation, in particular key-fold cross-validation or other data splits, but can we rely on those results, because many of them retrieve only the performance metric, like the area under the receiving operating curve, and not the variance, which we should have also to compare, to have comparable models for our tasks. So if we can go in the next slide, we can see that only one study, as I said, did not report internal validation, and half of the study only reported the variance along with the performance metric. For the last criterion, conceptual reproducibility, also called replicability, this is the ability to reproduce the results under conceptually similar conditions. So, for example, in other institutions with similar patients. This is task definition dependent, and it is focused on the performance of the external validation, which is rarely performed in the ML development studies, and we also saw that in our reviews only 2 of 24 performed external validation, which also saw a drop in the performance. And to avoid this, the suggestion from literature is to use multi-institution datasets from multiple geographical area. In our review, only 3 studies used a multi-institution dataset, the EICU US dataset, while no studies integrated datasets from multiple geographical area, and this could be an issue. this is that's it for the part of the medical approach. Konstantina, if you want to present the statistic one.
Yes, thank you, Emanuele. So back to me, starting with the statistical, continuing actually with the statistical approach. As you understood, because in our review we are currently focusing on the predictive modeling and how models do the prediction through multiple factor analysis and predictor analysis. We and the rest of the research globally are focusing on causal inference and reasoning, which means that the main focus and the main diagnostic, let's say, performance must be assessed in new patients. I mean, we take old data in order to apply them to new patients, to new people. So this actually means we need to focus also on generalization of the model and analysis. However, in statistical approach, there are also some biases we have to take in mind. The few common ones would be the sample bias, the confounding bias, and the algorithmic bias, which actually is the result from the other two. And you will see why in a minute. In the sample bias, it is that type of bias when we make wrong selection of the population, the targeted population, or have selecting or asking the wrong and their characteristics are not, let's say, fair between one characteristic. The most common one is when women are less representative to researches than men. And the other one is the missing, let's say, the necessary response when we have the missing values and some people do not answer, or we are missing the answers due to reporting issues. This actually can be separated in three different types. The selection bias, which includes the correct, let's say, selection based on the inclusion and exclusion criteria we are having for the specific problem we are trying to target. And also includes the selecting frame bias, which is used to collect data which do not cover the entire population of our interest. We may have a small, let's say, frame. We need to sometimes open up the frame. The other type is the survivorship bias, which means when we say survival, we don't actually mean only death or survive. We actually talking about if people are leaving the study for any reasons at all, that most of the times has nothing to do with the study. Some people might feel sick and want to leave. Others actually die, but okay. So one of the main problems we saw in our research was that many studies actually made death an event as an ICU readmission. They equalize the ICU readmission with death, which is not very statistically correct, from my opinion. So the question here was, how are we going to take that as an event or as a survivorship bias? And the last one is the non-response bias, which also includes the missing values, as we said earlier, participants not responding due to ethical or psychological reasons. And the main thing here is actually why these biases happen. Usually we have inadequate data collection due to poor quality of data, because some countries do not have like third world countries does not have good quality of data, no good reporting platforms. And as a study from Tolera showed there was a shortage of data entry formats and delays in supplies that affected healthcare data quality in low-income countries. Also, the data pre-processing sometimes does not happen as it should because in machine learning approaches and AI modeling we have to have sometimes a structured, sometimes an unstructured type of data, but in both cases, because the data types are so many, like image, text, and tabular numerical, the cleaning and transformation must be done in a very high cautious way. Also, the missing value handling is not always correct because different types of missing value handling should be applied to different cases. And last but not least, the data imbalance and fairness, where some characteristics observed are not actually representative equally in the dataset. So the next bias is that of the confounding factor. Confounding factors are a major concern, especially in medical data, because what can happen here is actually hide a confounding factor can actually hide the actual association between the predictor and the outcome, or falsely demonstrated. As you can see here, there are 4 different outputs of this. Either underestimate or overestimate the association, which leads sometimes to the false negative and false positive errors. Also to show that there is no real association or hide the real association of the predictors with the outcome. Some cases that has happened is like in the study of RAMSPEC, that 30% of the prediction studies reviewed, interpreted, included predictor in a causal manner, but suggesting that modifying a predictor could improve a patient's prognosis, which is a very misleading outcome. We cannot assume causal association with the outcome when we have a lot of factors. Okay. Another study presented dementia risk scores included that the BMI is protective. Actually, let's say that the higher the BMI, the better the outcome, which is also a very false outcome. Also confounding, when we have confounders in our studies, this is also a poor research methodology for what we are doing. Unfortunately, in this case, machine learning is totally useless, let's say in quotation, because it cannot distinguish mediators from confounders or recognize bias. So we have to do it as developers, as statisticians, we have to do it for it. There are a few methods trying to deal with confounders. Actually, the first and most common one is assessing predictive models through cross-validation or accuracy with the AUROC and other sensitivity or accurate metrics. Also by the confounding, which actually is when you remove the confounder, but this is also very risky because the confounder can also have a positive or negative effect on the model. And by controlling the confounder through minimizing or stabilizing it in order to control its impact. Lastly, for these type of biases, these all merge or have an effect on the algorithm itself, as you can imagine, because the algorithms of an AI model actually are trained and are built based on the data. If those biases are not mitigated or are not controlled, we will conclude to bias the algorithms and inaccurate diagnosis, decision making. So all parties should focus on fairness of the data in order to have an equal treatment of the patients. One more example of that bias is the study from Colacci, which they actually studied 760 articles, reported that only 12% of those articles had actually made an algorithmic bias assessment, and through those 12%, the 75% identified a bias. So you can see how important it is to have an algorithmic bias assessment or to have a mitigation of the above biases. This can lead to many challenges. First, lack of clear standards of fairness, not equally treat all patients with the same factors, also lack of contextual specificity, as we said earlier, and last but not least, because of the black box nature of deep learning, we cannot see why these results occur actually through the AI model. The mitigation can actually focus on the pre-work, like the correct framing of the problem, as we said earlier, and so you see now the connection. In order to have a data diversification and representation, actually equality, and also to manage bias in data preprocessing through missing data handling, confounders, and so on. In the technical approach, we can eliminate bias during model development and validation, actually to build the model based on that biases to be minimized, follow reporting guidelines and updates, and also create an explainable framework of the model through many frameworks like Sublime and visual explanation, which is actually, again, Emanuele will tell you more about in the next slides.
Okay, so explainability will be the last concept that we will talk about, and there is a growing research area in AI that targets this issue, so to reduce the black box problem and to make the model explainable also to not only to the developers, so they can understand how the predictions are made, but also for the clinicians, so that they can trust better the model in their clinical decision making. So this area targets those explainable artificial intelligence methods, and we can see that the most commonly used are SHAP, followed by LIME and the GradCam, but we won't enter into details, but mainly give a framework to understand and choose which of the explainable AI techniques is best for our task. So there are 5 main domains that we can look at. The stage refers to the timing at which the explanation is made, so before or after the model development. Regarding applicability, the model can be applied to different machine learning architecture or to specific frameworks. It could give a global explanation about the overall behavior of the model, or can give a local explanation of single predictions, how are made. Regarding the form, it can be rule-based, so we can have clear conditions that lead to specific predictions, or we can have a visual representation. For example, we can have the tumor area highlighted in a medical image. Finally, regarding the type, the most used is the feature important ranking, so features are ordered by the impact they have on predictions, and we can give an example of the most frequently method used. So the sharply additive explanations that assigns each feature an importance value, that is the Shapley value, for a particular prediction task. This is a post hoc model agnostic technique that can give both global and local explanations. It is rule-based but can also give visual representation and leverages feature important rankings. In our review, we saw that 2 of all the studies included used an explainable AI technique. The first, and it was the sharp one. The first found that the key predictors for the ICU readmission prediction task were peripheral oxygen saturation, respiratory rate, and heart rate, while the last gave more importance to severity scores, but it was trained, the model was trained on a specific ICU population of patients with the diagnosis of heart failure. So we can conclude with the same question. So should we trust AI predictions? So should we trust the results of deep learning development studies? We saw that many of them still have high risk of bias, and this is not only due to reporting issues like incorrect or incomplete reporting, but also to lack of reproducibility and generalizability of those results, which limit their clinical applicability. Even if we achieve the aforementioned properties, we have still a lack explainability, in particular of deep learning models, that can reduce the trust of the clinician on those models and also their applicability. So we truly think that the research team in this area should be heterogeneous, but in a positive way. So medical staff, researchers, and developers should be all involved in the early stages of the prediction model development, so that we can achieve results that are reproducible, generalizable, and also explainable. We conclude here our presentation, and thank you all for the attention.
Thank you very much for your presentation, Konstantina and Emanuele. Question, please. Well, if there are no questions, then we are going to take a short break. But before this break, I would like to say that the good news is that Sofia Rocha achieved to join our webinar. The plane problem was solved, happily. So she will talk after the break. Also, during the break, in case you did not register to our webinar, I say again that the registration button is still open, so please do it so. It's important for us and also for you to get the announcement about the slides and the registration being available on the website of the seminar. Therefore, we are going to take a 10 minutes break, so we are going to be back at 11:37. Okay? Thank you very much for all your presentation, and stay with us, get a good coffee, and be back in 10 minutes.
Okay, the recording has now started. So next off with our presentation, we have Sofia Couto da Rocha, Head of Innovation and Head of Virtual Client at Lusíada Saúde in Portugal. Please, Sofia, the floor is yours.
Hello everyone. Can you hear me well? Sorry, because I'm in a room that is a little noisy. First of all, I'm really sorry for that delay. I was in another Congress speaking in a table and our colleague just got delayed, so I had to be there stuck. So I'm really sorry, and sorry for the organization as well, because this generates trouble. So I didn't mean to generate any anxiety on that. So I'm trying just to share the best part here. Okay, here it is. Okay, so I'm going to try to be short, the best possible case. So I'll decided to bring, to start by explaining a little bit how I work in this field. So we use synthetic data for quite a while in different contexts. Mainly recently, we've been working in synthetic data for aging clocks. So I dedicate clinically to longevity and regenerative medicine. So it's very interesting nowadays that with the data that we have, mainly longitudinal data, that we can have these type of conclusions and informations based on, for instance, the age of an organ or the age of an overall, biological age of an overall human being. Let's think about DNA methylation, or let's think about how telomerases are at the moment. And with that said, we normally used before, not of course synthetic data, but just using data that we have to identify some variables and some mimic according to those variables, that we could take conclusions over that, which has been an interesting challenge. But at this moment, when data is more and more needed, and many times to have a conceptual digital twin of our patients, it's quite hard. We need to explore new fields, and that's how synthetic data entered our life. Just a note that probably for everyone that is listening today is not something new, because I assume that almost everyone here will work around these topics. But the thing is, data is not so as many as we thought. So there's a huge discussion in the last 7 years that there's a lot of data, and we are generating a lot of data and saving a lot of data, and most of the times we don't even know why or for what. But the thing is, when we look to open AI models, or when we look to large LLMs, we understand that whenever we are talking about accuracy growing, we know that we're reaching a plateau that is a little bit hard to move on, because the quantity of data starts to have incredibly higher. And that's another driver for synthetic data. Sorry, I think. Nice. Thank you. So what we've been doing lately is to improve this, to use more this artificial generated data that mimics, of course, statistical properties, real world, without revealing actually patient information. Because when we talk about this longitudinal approach, or these aging clocks, I used this first, I informed you in the first hand of that, because it's where we have the highest level of ethical concerns and difficulties. And so to address the privacy concerns and the data scarcity, we ended up starting to use synthetic data. It's super interesting in healthcare, of course, because it enables us to develop and test algorithms without compromising confidentiality, without compromising any issue regarding ethical conclusions or ethical secondary use. And of course, it's much easier to share across institutions than what we can do with real data. There's, of course, a super central concern, which is we have a lot of benefits, but there's, of course, a problem on perpetuating or even amplifying or generating new bias, which in the original data sets, they are, of course, already there according to different institutions and different focused in terms of research targets. And that is, of course, a problem that we need to address. So there's a growing reliance, of course, in AI in healthcare for diagnosis, for aging clocks, for treatments, planning, and so on. But there's also a huge importance on how these systems are only good if the data that I'm feeding them, it's trained, it's good as well, and they are trained in good models. So this idea on how we can amplify data without amplifying distortion is, of course, a huge challenge of these moments. And we all know that we dealt with bias all our lives. So everyone that did research knows that bias was something that we should be aware of, but nothing totally new. The only problem now is that we have the capacity to amplify it, and that's the point. We know that, of course, AI in healthcare is totally data-centric. And data rarely or probably never is neutral, and that is a challenge that we've been facing all the time. We also know that the big promise regarding synthetic data is the capacity to boost privacy or to keep privacy, to boost volume, and to have this scalability that is impossible with other type of data sets. But it is also true that we have this bias replication that we need to be aware of. So it's like when you think about synthetic epistemology, it's like we would have this variance and replication of all the doubts and the questions that we have with a very specific, and we have a group that just discusses ethical concerns with a very specific topic, which is before in the normal data sets, biases, we were, our humanity linearity was able to understand and identify those biases. And now, of course, as we are scaling up so much, we are no longer capable of doing this in the same capacity and impact. So technology is exponential, and we cannot, most of the times, even with really good auditable and explainable AI methodologies, it's quite harder and harder to do it. Not that I want to deep dive a lot on here, because I know that probably the people that is assisting today are more aware of these topics, but as you know, we can use different ways and different structures to generate synthetic data, or to do synthetic data. In our case, we use mostly 3 of them. So the generative adversarial networks, known as GANs, we use 2 neural network, a generator and a discriminator, to produce data indistinguishable from real data, theoretically indistinguishable. And then we use VAEs as well, so variational autoencoders, which encode data into a latent space and then decode again, and so we generate new data points, depending on the type of data that we normally want to generate. And then the agent-based models as well, which simulate interactions of agents to access their effects on the system. And this is super interesting to double check the synthetic data generated as well with the other 2 techniques. So there's a lot of potential pitfalls, as we already spoke, but to be more precise, for instance, GANs, which are extremely interesting, and they are the ones that we are using the most, may produce limited diversity because, of course, as minorities are already minor in the original data sets, they tend to ignore these minority classes. And for instance, when we talk about how powerful they are, GANs are super powerful, probably one of the most interesting ones at the moment. They can suffer from this collapse because of the lack of diversity. And whenever we want to identify connections, it's extremely, or variables that are connected between each other, covariance, for instance, it's extremely hard. Then the overfitting, which is again another problem. Synthetic data will replicate all the noise that we have in the original data and all the abnormalities. And sometimes the data sets are so large that even when we use other algorithms to review them and to normalize them in the good sense of normalization, not the mathematical one, in this sense, this is a little bit hard. And with these 2, we have this loss of dependency structure whenever we think about loss of correlation capacity, because relationship between variables might be impacted, and so they are not original and they make it hard. I brought an example of an open source, data set that probably all of you might know, because of the bias amplification pathway. This is super important to be focused on them. I'm going to go for the example after, but we know there's proxy bias. They are probably the most common ones. They are normally encoded in the real world variables as well. They are normally proxies for places in the world that people live, for ethnicity, and so on and so forth. Then structural bias, they are probably the ones that we hate the most. They normally reflect underrepresented subgroups or types of data. Then the latent representation distortion, which normally encoding spurious patterns has essential features. That happens a lot with GANs and VAs. So meaningless but frequent correlations end up being super expanded. And then this feedback loop amplification where there's a recursive distortion as we amplify the over and over again these bias. To give you a more tangible version, because I know these type of examples are very sky placing, we have, of course, solutions that are some of them better in some specific contexts and vice versa, other better in other ones. But I think probably one of the most interesting ones and basic ones and kind of La Palice output is balanced sampling, which not always, and whenever we talk about longitudinal data about patients, when we think about this aging clock, and that's why I gave this example, it's so interesting. It's extremely hard to have this super balanced sampling because we don't have so many patients with the same, the high quantity of data that we need with the different variables. So think about the life of a person that we followed from genetics to all the disease that they have, all the moments of different methylation of DNA along life, and then the outcome of their life, so how it ends or how it approaches to the end. Our N many times is equal to 1 in the sense that the more I want to be precise, the more I reduce my sample up to 1, which is a super challenge at this moment. I moved on because I forgot to share with you. But this example, another example that is more concrete and that you all can replicate it at home, which I think it's always a very interesting thing in this type of technologies to do it by ourselves. So I don't know if you all know the MIC third data set from MIT. I don't know if you can react here to tell yes or no. So it's a freely accessible data set. It's online with, of course, 40,000 patients data from ICU. They were admitted in the Beth Israel Deaconess Medical Center and in Boston, and from 2001 to 2012, so 2012. And it's super interesting because you can access this data set to actually do any study that you want. And imagine that you create a scenario about developing a mortality prediction model using synthetic data generated from this data set. There's an issue in the general data set because there are some ethnic groups like Hispanics, for instance, that are underrepresented due to the place where it was collected. And so the synthetic data that is generated without addressing this imbalance, imagine that you did not use any methodology to address, leads, of course, to a model that is that incorrectly predicts mortality risk different in these groups, as they do die less because they actually are super underrepresented. And that seems super basic failure, but it happens. And if you do it if you mimetize this is extremely interesting. So we would have to have reviewing this bias before, of course, going to any conclusion. Going back here. So I don't want to take a lot of time on the current solutions because honestly they are not as interesting that they should. Mainly many times we end up doing post-generation reweighting, which is always something that is not desirable, but it's still something that we are trying at the moment. One of the largest problems that we are, I think, near to solve is probably this lack of standard audit protocol to post-synthesis. So we review, but many times we don't know, like in a basic way to say, we don't know specifically to where we should look. And this is normally too late then to be able to reverse the problem. So then there are some issues regarding also the responsibility in the usage, and some of them might be very discussable, and it would be really interesting if we could discuss after, but there's regulatory ambiguity. We don't know what the FDA, EMA, or MHRA are thinking about. There's a lack of policy huge in synthetic training data. Of course, there's already a lack of policy in LLMs in healthcare. How couldn't we have this lack of synthetic training data policy? We don't know who's liable on harm occurred, because many times we already have this challenge in medical devices. Whenever they are parts of the medical device are built and trained from different players. Then there's this ethical distortion. We validate synthetic data outcomes as if they were clinically truth. So it's super interesting because, of course, patients can sue, but the ones that we use for the synthetic data, but then, of course, the real ones will suffer, as we don't know if it was probably grounded or not. So just to leave you some steps and some proposals on how we actually are tackling that and how we can look at it. So we try mostly to support audits, improve pre-synthesis audits. So many times we try to review the data sets in a way that we reduce, this is the obvious, but reduce this bias previously to generation. Then we use some constraint aware, we are constraint aware that GANs and VAs are going to be amplifiers of anything that we don't want them to. So we try to have metrics of fairness built in. Most of the times we don't use just one, so we overlap them. And then, of course, post-synthesis real-world validation. So the thing that I was saying, we use synthetic data from non-real patients applied to real patients' data sets that are from different contexts or from different sizes, and then we try to see how the reaction in the board's outcome is. Then transparent reporting is something that we normally, we end up identifying. We are the first ones to identify our bias so that we expose them. And if someone else wants to try the same path, to be aware already. And then, of course, any stakeholder inclusion with support. Like this example that I was giving you with MIC, any doctor that would work in a real-world environment in an ICU would know that Hispanic ethnics wouldn't have this difference in dying, but that would be an issue that sometimes when we talk about a lot of data, it's extremely hard to be naturally aware that the output didn't make that much sense. So just to reach my end, I would like to leave some overall messages. I know this is a very specific topic and it's a lot of information to share in 15 minutes, 15 to 20, minutes. But to leave it on the top of the table, we need to think about bias detection. So implement tools to identify and measure bias previously to in our data set, so that previously to generating synthetic data. Then the fairness aware algorithms. So using algorithms that were designed to maintain fairness across different demographics, groups, different ethnics, and so on and so forth. Then diverse stakeholders involvement. The more that we can involve different stakeholders, the more we can have them, and the more eyes we have and sensitivity to look at the data and think that it doesn't make sense. Then continuous monitoring is extremely important, mainly in this type of work, like I was sharing with you, like it is in aging clocks, because normally data is a longitudinal data that we collect for years and years and years. And so that while synthetic data has this potential to actually democratize, if you allow me, data access, it's crucially to proactively, of course, address the biases in firsthand. It's also crucial to think about this topic of the fairness algorithms. This is probably one of the best ways to face this challenge about synthetic data biases. And it's, of course, our responsibility to design all of that with accountability. And I feel that the more and more we talk about this, the more and more this type of groups of people that work with this are aware that we are all accountable for the outputs that those amazing technologies are putting in the world. And then do not, of course, trust the synthetic illusion of fairness that we face in most of these outputs. Many times we look to the output of the synthetic data and it sounds amazing. Please redo it and redo it again so that we can build models that are actually clinical truth and not just answering our outputs. So thank you very much. I know this is a very intense topic. I hope it was interesting for you all as it is for me.
Thank you. Thank you very much, Sofia, for this very insightful presentation and also for the inspiring quote that you added at the end, which I think can be a very nice point of discussion or of debate. So if anyone has any questions for Sofia, you can raise your hand, of course. If not, I do have one small question. Since you mentioned a regulatory ambiguity, I would like to ask you if you were thinking of shortcomings that are present in national legislation, like in Portugal, I assume, or at a more vast level, like European level.
Thank you, Sabrina. So honestly, I would love it to be in a national level, but it's not. I think it's in a world level. It's a super challenge. We are still struggling, not regarding synthetic data, we are still struggling in generating regulation that to LLMs, for instance, apply to healthcare. The more autonomy we give them, the less we know what to do with regulation. On the other hand, if we apply a lot of regulation, we are going to block innovation, and it turns to be just able to really large corporations, because it's super hard to handle regulation as we have in medical, in drugs, for instance. Normally only big corporations can handle it, as it's so deep and with a lot of steps. So what I'm hoping is that we can grow together in this type of groups that are super aware of the thematics and generate good practices guidelines, because it's much more interesting than really tough regulation. There is not going to cover everything, and there will always be a problem there. So even with the European AI Act, synthetic data is still somewhere there. So of course we can talk about how this data will impact and how algorithms will impact and measure things that are crucial or dangerous or that imply the highest level of safety. Still, it's not clear about synthetic data. So synthetic data normally turns to be in a sweet spot, which is, this is not real world data, so I can do whatever I want with it. And when we think about research and the outcomes, the real world outcomes of research, that's not true. I can use fake patients theoretically, but the conclusions and the impact is in real world patients, real world people. So I think this is a crucial thing. We need to include the synthetic data there. On the other hand, when we work with algorithms, it's extremely interesting to have these open options of synthetic data. I can play around with it forever and whatever I want. So I think this balance, and not to talk too much, but this balance between this freedom and responsibility, I think we have a lack of definition on that still.
Okay, thank you very much. Yes, it's surely a challenge between regulation and innovation. Finding a balance is always a challenge, but I think, that's something we can achieve progressively, especially at European level. So are there any further questions? Thank you, Sofia. Thank you. Any further questions? Otherwise, I think we can move forward. Also, just to remind you all that we are going to have a session for debates and discussions and Q&A at the end of this webinar. So if you have questions, please do not hesitate to raise your hand at that point. So our next presenter is Chiara Bellatreccia, who is a research fellow at the University of Bologna. Please, Chiara, when you want, the floor is yours.
Thank you. Okay, good morning, everyone. Thank you for being here. So I will try to share the screen. Just wait a second. Okay, can you see the screen? Can you confirm?
Yes, we can see it.
Okay, perfect. So today we're talking about addressing bias and data scarcity in AI-based skin disease diagnosis with non-dermoscopic images. This was a work that I did in collaboration with the Equitas project with Andrea Borghesi and Roberta Calegari. So in particular, we are going to introduce the problem. The problem in this case was the imbalance in the skin tone attribute in dermatological datasets, a very well-known problem, unfortunately, which can lead to possibly biased classification. This classification leads to a worse performance, especially against the darker skin tones, which are typically the underrepresented ones in the datasets. So the goal of this project was to augment a non-dermoscopic imbalanced dataset, which I will be introducing later, by using synthetic images generated by fine-tuning a stable diffusion model with Dreamboot, and to compare the classification bias between the augmented and the original dataset. This was done by using two different classifiers. The first classifier was a convolutional neural network, which is very widely used in dermatology, and the second one was a swim transformer. I don't know if you're familiar with the swim transformers, but they are very useful transformers for this kind of objective because they can capture very long-range dependencies with a low computational expense. So they are very ideal for this kind of situation. So this is an illustration of our pipeline. You have the original dataset, which again is composed, as I said, of non-dermoscopic images. What does non-dermoscopic mean? It means that essentially the images are taken with consumer-grade cameras, which are, for example, phone cameras. So they are not professional cameras and they are not the dermoscopy, which is that instrument that medicians use to diagnose, for example, skin cancer, things like that. This means that images are very, very different from each other in terms of illumination, in terms of areas which contain the disease, and obviously also the skin tone. So we needed in this case a pre-processing algorithm in order to have images of the same size, because in order to fit them into a model we need the images to have the same size. So for this reason we used a cropping algorithm, which is the first stage of our pre-processing, and after that I will delve into the pre-processing and the other stages later, but after that we trained both the convolutional neural network and the swim transformer on the original but processed dataset. And we also performed some kind of augmentation, as I said, by using stable diffusion and Dreamboot. After the augmentation, we trained the same two classifiers on three different augmented versions of the dataset, because when you generate synthetic images, again, you want to ask yourself, how many synthetic images should I augment the dataset with? We don't know a prior which is the best proportion for augmenting a dataset with the synthetic images. So in this case, we tried three different proportions and we trained both the classifiers on three different versions of the dataset augmented according to three different proportions, and we did a comparison of both the performance and fairness of the classifiers. So I will just have a short introduction of the dataset. The dataset is again a non-dermoscopic dataset of about 8,000 original images of about 273 pediatric patients at the Sant'Orsola Hospital here in Bologna. These people, children, had about 9 possible skin diseases. Obviously, as I said, the images are taken with phone cameras, so they show high variability in illumination, high variability in size and quality, inconsistent focus on affected skin areas, which is a very important problem here. Blurriness, so some images are blurrier than others. They actually contain maybe the disease, but we cannot see it very clearly. Imbalance in skin tone, and last, imbalance in disease class. So a lot of flaws in this data set. So as I said, the first step was the preprocessing step, in which we applied a sliding window-based algorithm, which employs the binary mask of the image. So it starts from the top left corner of each image and considers a patch in that image and computes the binary mask. And in particular, it computes the disease coverage, which is roughly a measure of the contrast in that patch. Because obviously, if there is going it's not exactly the contrast, but it's actually the ratio between the positive and the zero entry of the binary mask. We will acknowledge whether the skin disease is present in that patch or not. And so this is a very useful way to detect whether the disease is present or not. If the disease is present, the crop is taken. If it's not present, the crop is discarded. So the result is about 330,000 crops of uniform size. Not all crops are good crops. So you can see here I put the essentially worst crops that we had. Some crops show very poor illumination and blurriness. Other they are shadowed. Other, they show edges because the algorithm is not sensible to this kind of features like edges. Others show just body hair and nothing, or unnecessary body parts. And also here we have, for example, blurriness. So essentially, they said that there is a lot of noise in this data set, even after the preprocessing. That's why, for example, in a later implementation, we are trying to address this kind of features that are generated by the algorithm. So after that, we had to estimate the skin tone because, as you may know, there are very, like in this case, we did not have the labels, the official skin tone labels from the medicians. This is a problem to estimate, as I said, the bias, because if we want to estimate the bias of the classification on different skin tones, we need to measure the skin tones. Unfortunately, we didn't have the official label from the medicians. We are, again, working on that. We're developing a web application in which the medicians can insert the exact skin tone of each patient. But at the time, we only had an automated way to detect it, and we chose the individual typology angle, which is a very well-known metric in dermatology to measure the skin tone, and we obtained from that the distribution of skin tones. So we can see the 6 different skin tones according to the Fitzpatrick scale, and we can see the different proportions. You can see that the dark and brown skin tones are the minority in our data set, while instead all the other skin tones are somehow well represented. So then we move to the augmentation part, for which, as I said, we chose the fine-tuning of stable diffusion via Dreamboot. We had to manually select the images of dark and brown skin tones, and we built some mini data sets on which we could use the Dreamboot technique to generate new images using stable diffusion. Of course, we performed some research to find the set of optimal hyperparameters, and then we selected the best models to delve into essentially the synthetic augmentation. Okay, here are some of the results. You can see for the 3 different diseases that we chose. Another important thing is that we had 9 different diseases in total, but we could choose just 3 diseases to augment. This is because for the other 6 diseases, we didn't even have enough dark and brown patients to start with. So this is another kind of problem, which is we had a very underrepresented data set, so it was very difficult. But you can see some of the results here for the 3 diseases for which we had some kind of dark and brown patients. And then we come to the performance and fairness comparison. So for what regards the convolutional neural network, as I said before, we compared the performance between the training on the original process dataset and the training on 3 different versions, 3 different augmented versions of the dataset by using, as I said, synthetic data. For what regards the convolutional neural network, the best approach was the so-called AugMax approach, which comprised essentially the highest amount of added synthetic images. So this means that the convolutional neural network worked better when we added the highest amount of synthetic images to the dataset. In order to assess the bias, we use the three fairness metrics, which are disparate impact, equalized odds ratio, and predictive rate ratio. These were used in a similar paper, which did a very similar thing to ours. So we based our decision on the literature which was already present, and we subdivided, again, looking at the literature, the skin tones in a minority group, which comprised the dark and brown skin tones, and the majority group, which comprised the other more common skin tones, which were already correctly represented. So you can see here, by using this convolutional neural network, you have on the left, you have the results on the original data set, and this gray stripe essentially is the so-called fairness zone, which is the zone in which the three different metrics should ideally be found, actually. So you can see that a lot of fairness metrics are just out of the gray zone. Looking on the right, you can see instead the performance on the augmented dataset, in this case on the augmented dataset with the most amount of synthetic images. You can see that some of the metrics actually approach the gray zone, meaning that they approach the so-called fairness values. In particular, 17 out of 27 fairness metrics values improved, even some of them obviously slightly, but they show the sign of improvement, which is the most important thing. In the case of the convolutional neural network, the performance also improved remarkably. This is mostly because of a regularization effect that the synthetic images had on the classification. For what regards the skin transformer, instead, the best approach was the one with the Agmin approach, which essentially provided the less amount of added synthetic images. This was mostly because the swim transformer was pre-trained, and so there was less room for improvement here. Okay, so we can see again the results on the left on the original dataset, on the right on the augmented dataset. You can see that even in this case we have a very comparable performance, like slightly worse on the augmented dataset, but it's very much acceptable. And then we have on the left all the fairness metrics, which are widely outside the gray zone, and on the right we can see that some of the fairness metrics values approached the gray zone. So in particular, 15 out of 27 fairness metrics values have improved. So even in this case, there was an improvement in, especially in fairness. So in conclusion, the results demonstrated that synthetic images in this case have a positive impact on classification, both in terms of performance, in the case of the CNN, and fairness. The improvement is more remarkable for the convolutional neural network, again, mostly because the convolutional neural network was trained from scratch, while instead the swing transformer was pre-trained. There are several improvements to the pipeline. The first one, which we are working on that, to improve the processing to enhance the classifier performance, because as I said, this dataset has a lot of noise. We're actually trying to improve the processing and later to also maybe make the dataset public, because right now it is not public, but we want to publish the dataset obviously in a widely anonymized version. So the crops are more or less anonymous if we take out the crops which contain some body parts like noses or eyes and things like that. The second room for improvement is to generate dark-skinned images starting from light-skinned images. So as I said, right now we started from just dark-skinned images to generate synthetic data. But we could in principle start from the light-skinned images, but we have to pay very much attention on that. This is because the different dermatological diseases can have very different textures on different skin tones. So just generating dark-skinned images, just starting from light-skinned images and, for example, colorizing them with the different skin tones could be not enough, because sometimes we also have a different texture. So it's a very complicated problem. And another thing is that as I said, this is a non-dermoscopic dataset. So again, we have very high variability in different skin tones and features. So this is indeed a very complicated field, but we hope to work on the right direction. So I thank you for your presentation. I will stop sharing, I think. Okay, right here. And if you have any questions, I am here to answer.
Thank you, Chiara. Thank you for your time and for your comprehensive presentation. This research you're conducting is very, very interesting.
Thank you.
Yes, if there are any questions about this for Chiara. Okay, I believe there are none. So we can proceed with the next speaker, who is Panagiotis Tsakanikas, senior researcher at the Institute of Communication and Computer Systems in Greece.
Yes, hello, Sabrina.
Hello.
Hello to everyone. Have a good, a good day. Thank you for inviting me to this insightful webinar. So let me share my screen. I will present you a framework more or less about bias assessment. Sorry, just to find the right one. I think this is one. Okay, I hope you can see it. Let me put it in the... Okay. Let me go to this beginning. I'm trying to go to the beginning, sorry. So, as I'm going upwards, I will today I will speak about a module framework that we're building in the context of project European project that I'm working now. Is the OncoDIR, and I will talk about the building AI trust through bias identification. Some words about the OncoDIR is a project that will finally it will develop and provide a platform, a platform based on artificial intelligence in order to provide recommendation services based on the input from the citizens, from the policy makers, and also from the clinicians. So it has a lot of, let's say, a diversity of the input and the data that we have. It has from several, inputs from several stakeholders of for colorectal cancer. And we also consider here in this project factors such as lifestyle, nutrition, and economics. Concerning the bias, we think that the systematic unfairness in AI models due to the data and features or algorithms that will be used and developed within the project is a crucial issue that should be mitigated and also reported to the end users in order to have the human in the loop as it necessitates to be. I will start with a brief introduction. There are several types of bias that has been commonly recognized in the AI. The bias of the data per se, when the training data that we have are not representative and not reflect the real world scenarios. Also there is the algorithmic bias where the machine learning models and all the tools that the developers are creating are systematically unfair to several groups in the healthcare domain. The label bias, where the labels that are used for the training carry human prejudices. So this also affect implicit bias both in the data and also in the training of the models. And finally, it's a huge, let's say, issue around the hidden bias, where hidden dimensional patterns in the data create unintended interference, because it's very difficult from the humans and the developers to see if there are any bias in several dimensions of the data. Then they overlooked it, and they are going to search why the algorithms are unfair. So in the project, the application that the framework is going to be applied is for the misclassifying conditions due to the bias training data, for the recommendations that the tool platform of the project will create, the bias into those recommendations, and also for the public health data is the under prediction of disease risks in specific regions. In the framework of the tool, we take under consideration the bias that is or maybe in individual features, the high dimensional data that can hide complex biases, and it's very difficult for someone to see beforehand. And also we employed several techniques like anomaly detection and sub measures in order to have also the explainability and see other types of bias that are not being detected by the two that I mentioned earlier. So the insights in the real life in order to based on the bias that is into the data and the algorithmic amplification of those data is to have better patient outcomes in order to avoid misdiagnosis or recommendations of incorrect treatments, to ensure that the models and the prediction tools that don't favor one group over the other based on either a feature or even a set of features, to enforce and promote the trustworthy AI, and also to have a regulatory compliance for the systems, since it's very sensitive in the healthcare domain. What we found during the development, which is carried on nowadays also, is that we need a multi-phase detection approach, starting from the quality assessment of the data to traditional bias metrics that has been used, moving towards the high-dimensional data detection, bias detection. We also move towards the community detections in order to see small communities that may indicate biases over specific groups of populations, and we provide and integrating into this framework the explainability via the sub metrics, and also we employed autoencoder-based hidden bias detection in order to find and report to the developers and also other end users like clinicians and policy-making actors about the situation for the bias in both the decision and also the data. Here is a workflow of the diagram, a workflow diagram of this multi-phase bias detection framework, which, as I said, we go through it very quickly, from the data pre-processing profiling of the data to the traditional bias metrics that is being used, integrated with high-dimensional bias detection, sub-based bias detection and explainability of the biases, and also to the community bias detection towards the final scope to alerting for the biases and create reports. We report the outputs of the tool is a PDF for the end users, and also a JSON file format for the developers in order to incorporate it into their tools. So this is an example of how the JSON is, and for the PDF report screenshot, we have several reports. As I said for the previous slides from the workflow, each one section represents its workflow and the bias that has been detected therein. And we also use some color formatting in order to easily for the users to see where are the most severe bias and which are the interconnections, the correlations. And so I can go through very quickly more detailed, let's say, for each step of this multi-phased approach that I'm happy to see that we are introducing. This is the data quality assessment where we look about imbalanced data, missing data, skewness and kurtosis, coefficient variation and other metrics for the quality of the data, which are important and leads to give indications about the bias that may be inherited in the dataset that we have. Several distributional based metrics for the bias detection have also been used and employed in order to detect the bias for each feature of the dataset that we have independently. And then those datasets, those metrics are used in combination, and we have a dynamic, I will show later, dynamic scoring system for depicting the most severe biases that have been detected in order to alert the end users about those. In the high dimensional data detection, we perform dimensionality reduction in order to have pseudo features that comprise from combinations of features that we have earlier in the datasets. And then we have some clustering algorithms in order to detect those hidden bias. As we can see in those charts, depictions here, the separation of two classes suggests that the model might rely heavily on certain patterns of the data. If one cluster is predominantly from an overrepresented group, like this is for the case of the younger individuals that we have in this distribution here, is shown much clearer. Excuse me. The model will favor towards that group and will lead to biased outcomes as we expect. So high dimensional data is very crucial, and to further strengthen this approach, we introduced and integrated the anomaly detection, where we found several through the clustering and the distribution of the anomaly scores that we calculated on the datasets. We can find certain groups that appears as outliers, although they are not, or they are not easily seen by the developers mainly. And those datasets, those smaller groups, those smaller subgroups of people may be, or may they for sure be underrepresented in the dataset and leads to biased decisions over this subgroup of people. Next, for the explainability, we used this APPO metrics. To do so, we used autoencoders in order to learn the feature representation. And though the data from those autoencoders, the reconstruction error that we get as a result can be used as an anomaly measure where higher errors suggest potential bias. This means that we don't have enough data to have a low reconstruction error, and this means that for these latent variables, the model, there is apparent a bias intercorrelated with those latent variables. In order to interrogate more this, we use the XJ-Boost in order to predict the error levels and produce the explainability analysis over the sub-values for each feature, and we create the sub-feature important plots where we give to the end users, as you can see here, we give the user the most the features that have the most significance towards the bias of not for the prediction but for the bias that has been inserted into the model, or the bias that has been most influence that is most influencing the outputs of the models. So the developers can go back and retrain and adjust their models or the datasets. In terms of the community bias detections, we employed graph-based clustering in order to detect some systemic bias that may inherit into data. So for this reason, we produce graph representations in order to show the interactions and the relations between the several subgroups, not the subgroups as we see the datasets, let's say older vs young, but in a higher dimensional space. To see and inform the end users about those biases and those homogeneous or unhomogeneous communities that we have, in order to be acknowledged of the biases that may be within the dataset, and also will be included or integrated through the modeling and the prediction procedures to the end users like the policymakers or the clinicians. As I said earlier about the automatic bias thresholding, from all those metrics that we calculated earlier, as I saw earlier, there is this thresholding for the mild, moderate, severe, and very severe bias, and the recommendation about monitoring, investigated, or any critical action that may be needed about the dataset and the algorithm that has been also employed. Those bias, those also thresholding triggers and reports into the reports that I have shown you earlier, the alerts about the bias for the users to be acknowledged and take the necessary mitigation actions. So for the insight, some insights for the end users, as an example, and for the usefulness and the benefits for the bias assessment in general, not only the one that I'm presenting here. Let's say that we have a cancer risk prediction model, which is supposed to support daily diagnostics. If it uses demographic and lifestyle data to estimate individual risk levels, the bias assessment revealed that the training from the previous graphs that I have been showing, that the training data have an overrepresentation of the individual, younger individuals under the somewhere about 30-year-old, and there is another underrepresentation of those for older people above 50. The bias indicators shows that we have a skewed dataset. From the PCI and the clustering analysis that we saw that the younger individuals form distinct clusters that influences the model outcomes, and also from the explainability, the sub-values that we get from the analysis, that age is one of the top bias contributing features in the dataset. So combining all this information from several aspects, the user can have a more comprehensive and solid justification and notation of the bias. As the real world impact, and on the basis of the end users, the patients and the clinicians, and not the developers from their perspective, the older individual will systematically have lower risk scores concerning the colorectal cancer, despite having higher actual risk. This will lead to a delayed or missed screening for the elder population, and also from the side of the population policy making stakeholders, and also the clinician stakeholders, their trust in the AI recommendations will fall down, and they won't even use them. Some benefits are from this assessment, from the bias assessment, is the early detection, so the fairness improvements, and the clinical trust, which is a very crucial and critical issue in the health domain in general about the AI tools that are used, and it's very significant to be regulatory compliant and support the ethical and legal requirements for equitable AI in the healthcare. That's all for me. I would like to thank you. And if you have any questions or concerns, mostly I'm concerned about the concerns. Thank you.
Thank you, Panos, for your very insightful presentation. Are there any questions? Yes, please, Adina.
Hello, thank you very much for your presentation. I would have two questions. First one, is there a risk of misattribution when sharp feature attribution that explain the model errors, they do not capture adequately the underlying bias in the original model? This would be the first question, and then I shall ask a second one.
This is a very, this concerns me a lot, at least for the development of these tools, because that's why we use this multi-phase approach, that we have supports for the bias that we have detected in several different dimensions and aspects of the datasets, from specific features to community-based, distribution-based metrics, and also for the errors of a reconstruction of errors and high-dimensional space for them. So all of those metrics are combined together and give the output apart from each one individually for its aspect, either high dimensional or one base dimensional data assessment. We combined the bias alerting system for all the aspects that we use. There are room for to be better on those, but I think it's a good first step towards this.
Okay, thank you. And the second question would be, obviously you give a very good perspective on the different aspects of bias in the data set they are using. But I was wondering if you also give them, except the analysis, if you give them some hints, I don't know if it is possible, on how to improve that. I mean, on how to reduce the bias. For example, use some more examples of this type, or use 30% more, or something like this. I don't know if it is possible. I just want to find out.
Yes, I thought about it to include some mitigation actions for the users, mainly for the developers. But I think that it will be very limited for them to how to mitigate their data. So we just report and showcase where the most of the bias is reflected on what features or what combination of features in the high dimensional space, and tell them that this population having those features or those issues concerning the bias and unfairness after model deployment, that you should look at those specific features that have those significance reported through the sub matrix. So there are a lot of mitigation actions that someone can take, and this I thought you are right. Maybe some mitigation proposals may be apparent, but I thought not limit its developer on how we'll introduce, because he might want to go back to the source of the data and ask for more data of some population that is lags behind.
Okay, thank you.
Thank you, Adina.
Okay, there is another question or comment by Juulia.
Yeah, hi. This is another question to the presentation, which was excellent. Thank you. I was just thinking, since I'm the last speaker in the program, and I think we're perhaps running a bit late on the schedule, and I have an overlapping meeting starting 1:15 So, so if you want to go through with the program as it is, I'm fine if you want to skip my talk, since I don't think I have time for it if we, there's one presenter before me. So just informing that.
Yes, thank you for that. And yes, we are running with a slight of delay, but if the next presenter, Andrei Olaru, is okay, we can switch to the two presentations. So Juulia, you can present now.
Problem.
Okay, if that's okay with you. Okay, I'm just going to, introduce you real quickly. This is Juulia Jylhävä, Senior Research Fellow at the Faculty of Medicine and Health Technology at the Tampere University in Finland. Please, the floor is yours.
Okay, thank you. And thanks so much for agreeing to change the slots. So, I'm a Senior Research Fellow and a Group Leader of Systems Biology of Aging. So that's why my take on AI and machine learning approaches is very much aging-oriented. And I look through everything through the lens of aging, biological aging especially. So today I thought I'd present a case example, what we've been doing and learning from our use with electronic health records, applying NLP models to it. So I'd first present the case example, the actual work that we've been doing, and then highlight some challenges and some fixes that we've mitigated some of the risks and challenges. So I hope this is a real study, so I hope you treat this confidential, even though I'm happy and fine sharing our data and findings. So with that, a bit of background. So we know that many clinically significant health conditions, they are frequently underreported as diagnostic codes, or they are only reported in the textual electronic health records. So only in the free text, whatever physician, nurse, physiotherapist has written about the patient. And then we also know that the systematic unreporting is particularly relevant for aging-related outcomes, so more relevant than the other outcomes. So for some reason, it seems to be underreported to a much greater rate in old age and aging-related conditions. So this is one problem, and then on the other hand, there is this problem that many conditions like mobility limitations, they don't have ICD codes. They are never reported as structured data. There are, of course, questionnaires, but not like universal code for that. And that's a pity because they carry a lot of relevant information on individuals' health that could be used in various purposes, but it's not accessible without these modern AI methods. So of course one can use manual abstraction, and that has been used, but then we all know that's not really doable when we have large masses of data. This maybe giving a bit of context to this is that this is kind of a issue because roughly 80% of all the electronic health records, they are textual data. So they're very low maintenance data, but they contain this wealth of information on our health. It's like a treasure trove in that sense. So in this study, we had kind of two aims. First, we wanted to pilot and put out this pipeline that we are testing. So we are using this deep learning AI method. We used natural language processing and named entity recognition task to the electronic health records, text records, to identify 4 very common aging-related outcomes: incontinence, falls, mobility limitations, and experiences of loneliness. So we wanted to first test the method, whether it can reliably identify these conditions, and then we wanted to, of course, see, okay, we can capture this and this number of individuals with these issues, but does it improve our prediction, and do the cases seem reliable that we can identify with NLP and so on. And then we, of course, wanted to compare our NLP-driven cases to those that are captured by ICD codes for falls and incontinence. So mobility limitations and loneliness don't have ICD codes, so we didn't have any benchmark to compare with there. So our data is real-world patient data from Central Finland Wellbeing Services County. We actually first, when we started the project, we thought that, as so many others, that why don't we just try with the mimic data, but then we realized that it wouldn't work. And we were lucky to get this real-world data quite quickly. So we have over 10 million free text records from a bit more than 100,000 individuals, and our records. So we wanted to ensure good coverage, representativeness. So they cover primary, secondary, tertiary, and institutional care from 2010 to 2022. And all the texts that were retrieved, they covered all medical specialties, so they were not restricted to any medical specialty. They also include reports and recordings from nurses, physiotherapists, social workers, basically everyone who has access and records some information about patients in this Wellbeing Services County. And then we, of course, had diagnosis and administrative data, mortality, other demographics, and so on. And our study group was aged from 50 to 80. And the gender distribution was quite well aligned with what we have in many of our other cohort studies. So a bit more than half were female. And that's quite representative because we know that female tend to use a bit more healthcare services than men. And roughly 20% of patients died during the period. So as I said, the method was the NLP based on NER, and we used Google's bidirectional encoder representations from transformers, so BERT model, and that was pre-trained for the Finnish language, so FinBERT. And then we identified these 4 conditions. And we used the kind of the standard procedure and pipeline. So we trained the model with manual labeling. So domain experts and people trained to detect the correct phrases, passages, labeled the text. And then we used the biotagging scheme and then assigned each health condition or issue, like yes, being present, no, no problem. In some cases, we assumed that an absence of a mention of an issue is actually absence of the issue, where it was reasonable to assume so. And then for mobility limitations, we actually wanted to test the model a bit more, so we made a scale from one is really highly limited mobility, split half is some mobility limitations or being able to move but with some pain, and then 0 is no mobility limitations. and standard evaluation criteria for our model. So precision recall and F1 score were derived and compared to a human rater. And then we, of course, took the prevalence estimates by age category and compared to the ICD-10 codes to see if what we come up with is reasonable, reliable, and as expected. And then finally, we did Cox regression model to assess the association with all-cause mortality using these NLP-derived cases and those that can be identified with ICD-10 codes. And this is just detailed presentation of the pipeline that we have. So we, of course, encrypted all the text so that all the names, addresses, phone numbers, and those, they were masked using regular expressions. And then we went through this pipeline of validating, testing the model, training it, and then looking at doing actual inferences of the data. And this is just a recap of the model evaluation. So precision recall and then F1 score, which is the harmonic mean of precision recall, was used to assess the models. And we kind of wanted to be here, of course, with our models in the very good range. It turned out to be a bit tricky for some cases, but then revisiting, going back to data, training a bit further, then it did some improvement to that. And we found that when we used the keywords to retrieve the relevant text, this was just translated from Finnish to English, just some crude examples, what they were like. We found that for most of these item categories or problems, there were like 5, 6, 7 key passages that identified above 90% of the cases who actually had this condition. So in this sense, these exemplar cases turned out to be, in a way, a bit easier than we thought, because we were so able to capture them with relatively simple passages and phrases. And here are some results. So we found very good F1 scores across all the models. We found, of course, as expected, well, not of course, but as expected, that our NLP model would capture more cases compared to the ICD-10 codes for falls and incontinence. So this was good. And then we, of course, overlapped or intersected what we found with NLP and ICD-10 codes. And we found that the overlap was not perfect, so that if we would only rely on AI, we would actually also miss something, because sometimes there are cases when only structured ICD code is entered. But then if we only relied on ICD-10 codes, then we would lose much more. So there's a balance in that. But overall, we were very happy with the F1 scores that were in the very good range. And then we checked the proportions of positive cases or individuals with problems across each category, and as expected, the proportions with individuals with issues and problems, they went up with age. So in that sense, this gave confidence to our model. And then we predicted mortality using the ICD-10 versus NLP cases, and we found higher predictive performance for our NLP model, both in men and women, and also in when stratified by sex, and also in the full sample. And for mobility, we were also happy to find that there was this dose-responsive pattern. So mobility limitations were associated with higher mortality, and also so that more severe limitations had higher hazard ratios than milder limitations. So then I'm going into the risks and challenges, and only kind of presenting the most pertinent ones to our example. There are many, but these are the ones that we can highlight as those that are relevant to this particular approach. So first is the temporal context loss. So whenever there's mentioning that patient, say they had a fall, we found that the model is actually very well able to capture the fall, but sometimes when there is a temporal attribute, like it might have been 2 years ago, so then it wouldn't actually represent today's situation. This was not a major issue in our approach because we were, in any effect, we were interested in patients' history. So if there was any fall at any point during their life, that was considered a risk. So the temporal context was not an issue here, but for other applications, it definitely would be. Although for falls, so we actually did a lot of checking, and we found that in majority of the cases where there was reported that the patient had a fall, it was very recent. So maybe in 1 or 2% of the cases, it was something that happened several years ago. And if it was mentioned, then it was somehow relevant to the person's or patient's present condition because it was mentioned. And then there is this attribution error or misattribution. So information that refers to someone other than the patient, so their spouse, child, parent, like let's say the patient's wife had a fall, and now this person who now came to see the doctor, they might need some home care or help because the wife who had a fall and is now maybe hospitalized is unable to take care of the patient. So in these kind of cases happened. Also, they were quite infrequent, but we saw those. So in this case, we would just get a false positive for this patient, and we tried some approaches to actually define the subject, but it turned out to be very challenging because these cases were so infrequent where it actually referred to someone else than the patient. So we decided to accept the low number of potential false positives to not to compromise the overall model performance because we didn't have enough training data, so to speak, to assign the subject because they were so infrequent. Then this was also a problem negation detection. So we know that NER is the NLP model that we used or task is noun based. So there are issues with words like no. So the model had severe issues in understanding word no. And we actually came up with a custom solution to this because it depends also in the Finnish language context. It depends on the trunk of the word. And this was pertinent to loneliness, not so much to the others, but loneliness when you express it. The person is experiencing loneliness or not experiencing loneliness. The trunk of the word is exactly the same, and sometimes the surroundings or the context can be the same. So here we came up with the custom solution to single out those false positives that were actually true negatives. And they were not many, but in the end, this approach worked out. And then this is probably a common problem to everyone working with textual electronic health records. So healthcare professionals like to use their sometimes jargon, slang, abbreviations, shortened words. And we noticed that, yeah, there are definitely these. But then we worked with domain experts and we captured all the most common abbreviations when they were unambiguous, so that they were specific to, let's say there is an abbreviation of MI, myocardial infarction. It can also mean something else. So there we were very careful that it was trained and detected in the right context. And same for the shortened words. So those were the major issues that we kind of still have to deal with, because this is what I presented is just an exemplar of cases, and we are actually building a full-scale model called Electronic Failed Index, including many other than these 4 items that I presented. So we still have to tackle and resolve some of these issues when we put out the full Electronic Failed Index model, which we are now working on. So with that, I'd like to thank all my group members and funders and collaborators who made this work possible. So, and I'm happy to take any questions you have.
Thank you. Thank you very much, Juulia, for your presentation. Yes, Andrei?
Yeah, thank you. So I have a small question. You said that on pre-processing you use irregular expressions to mask out things like names and phone numbers. Aren't names and phone numbers in their own like in their own fields? Can't they be masked out just by masking the whole field? Why would you need regular expressions?
So yeah, so the whole field like a column. So,
yeah.
In the data. So those were of course excluded. So and we had only dummy IDs. But then there are very often in the texts, the nurses or the doctors they actually mention the person's name, not surname, but usually they speak them with their first name.
How can you separate first names from like other things?
So we used from the Finnish population register, we used all the name information, address information there is, and then we created the list. So yeah. But I have to say that this worked maybe a bit too well because sometimes the masking algorithm had mistakenly taken out like some test results that were numbered that kind of bit resembled maybe phone numbers or something. So.
So yeah, because it's difficult to mask them in text, especially the names.
Yeah.
Okay.
It is.
Okay,
it is. But
are there any further questions? If not, I guess we can move forward. Thank you once again, Juulia, for joining us today. And okay. Moving forward, we have our next presenter, Andrei Olaru, which is a researcher and associate professor at the National University of Science and Technology Politehnica Bucharest in Romania. Please, Andrei, the floor is yours.
Yes, thank you. I hope you can see my screen. Yes?
Yes, we can.
So, hello, I'm Andrei Olaru, and I will present some preliminary work on building a framework for bias analysis in data in terms of helping researchers analyze bias, detect and mitigate bias in data. So I won't go over the terminology anymore. The definitions are definitely quite clear for everyone in this webinar. So we have bias, and we have things like data sets, data records, attributes, and classes, and we have protected attributes, and we have bias, and various types of bias, of which I have highlighted here: explicit and implicit bias, as well as the concept of fairness that has been discussed already before. So it is clear from all the other presentations that detecting bias can bring difficulties. So we have set out to build a tool to assist researchers and other people working with data sets in maybe identifying biases more easily. There are already some other frameworks, not awfully many. There's the IBM a Fairness 360, let's say, collection of tools, which is a great collection of algorithms and metrics. It's available in R and also in Python. However, it is offered as a collection of tools, so one has to actually go and see what algorithms exist, and one has to manage connecting the loading of data sets and algorithms and measurements by hand in a program. There's also risk of bias visualization, which is implemented in R, and has a web app which makes usability better. However, many of the machine learning tools, of the machine learning libraries, are now implemented in Python. So if one wishes to analyze algorithmic bias, for instance, in a model implemented in Python, it would be more difficult. Also, it is more difficult to add new algorithms and metrics without actually going into the library. There's also the Fair Models initiative, also implemented in R, which features a variety of plots which are very useful in evaluating bias in data. Again, there's the issue of interoperability with Python, and at least from what we saw, it's reasonably difficult to add metrics that have not that were not present in the framework before. So so what we found is that there are challenges related to interoperability, to extensibility of frameworks, and here also there's a lack of support for non-tabular data. So the initiative that I'm presenting is to construct a framework that integrates existing work, especially the IBM AIF 360 framework, that integrates things in a platform that is more usable for researchers, and also maybe for people who do not have a significant programming experience. So as end result, we envision a visual application, a tool to support the analysis of data, especially from the point of view of bias detection, which features pluggable blocks that can be plugged in the application, which would be algorithms for bias detection, algorithms for bias mitigation, also various tools such as attribute selection, filtering of the data set, so things that, for instance, pandas can do easily, but integrated in a more visual manner. Running predictions on the data set using algorithms or training models on a given data set. Also things like plots and the computation of metrics. All of these things exist already, so the point is to build a framework that integrates them, and so that they are easy to interoperate by someone without programming experience, for non-programmers. And the target is to build a visual language in which these blocks can be assembled in order to build processing pipelines. So something like this graph, where we can load a data set, we can maybe partition the attribute set in various partitions, compute correlations between attributes in the data set, also train a model on the train a model on the data, perform inference, correlate distributions between some attributes and the class that was inferred by the algorithm, build plots, and so on. And this can be done visually under two conditions. One, things need to match between algorithms and between methods. things like data types and dimensionality. And also another challenge is if we want to make this as a plug and play model in which it is easy to plug other algorithms, then there is a certain amount of standardization that should be achieved. But for instance, in IBM's framework, sort of this standardization already exists. So the algorithms generally have similar sets of parameters that they need to run on. So in this framework, the objects that we work with are data frames or data sets, sets of attributes, sets of labels, either ground truth labels for training or predicted labels predicted by the prediction models. Selections of record or data allowing for the selection of certain records in the data set. Models themselves, so the parameters of models, information on distributions and metrics. These objects would be transferred between various processes such as algorithms, model training, model inference, and the computation of metrics and distributions. And the results of these would be shown in plots. And also we have discovered while designing this that we also need some control elements, which will probably prove to be a challenge, because some processes need to be run several times. So we need the idea of iteration, and the results need to be aggregated. So we need aggregators to compose those results into one metric, or anyway a lower dimensionality result. Also, in order to make this more usable, we would need things from programming language such as object identifiers, so that one can identify like a data sets or parts of data sets or other objects that have been constructed, and a means to indicate flows of processing, like the graph that I showed you earlier. So there are several challenges here, but especially in the engineering part, we have experience from previous projects on how to dynamically work with machine learning models and other machine-related tools. So on the engineering side, there are things that are, well, they're technical, but they need to be solved when someone actually needs to run these tools. So things like package management, loading of models or of data sets and so on, and the interoperation of data types, so actually the uniformity of data types across a processing pipeline. In terms of usability, there are challenges in building a visual interface that would allow specifying the processing pipelines, challenges related to the dimensionality of data, especially for someone who is a non-programmer, to be able to understand that various dimensions of data need to match, and more programming related elements such as identifiers and iterations. So a preliminary very high level architecture based on AIF 360 as a backend for all the algorithm implementation is presented here. So we have an object storage for objects that are used in the processing pipeline, and elements to control the processing and to execute the various, to load and execute the various components that are needed in the processing pipeline. And this should be done, the elements of the processing pipeline will have to be interpolated sort of automatically in the back without having to, without the user actually having to write a program. Beyond the implementation of this framework, future items would be automatic variation of parameters, adding new features to analyze based on existing data, features that may not be present in the initial data set, interoperation with other frameworks providing algorithms or metrics, and an obvious choice would be to integrate LLMs that would help in interoperating components and in translating data between formats, which is something that LLMs can do quite well. I'd like to also thank our colleague in the ENFIELD project, Isel Grau Garcia from TUE, for contributing and giving us good advice, especially on the technical side of bias. Thank you, and your questions are welcome.
Thank you very much, Andrei, also for agreeing just before to switch and being the last speaker for today.
Sure, no problem.
So are there any questions, which I think we can integrate at this point with the session, with the last session, with the discussion, debate, or a general Q&A based on what we have heard and seen today? Yes, Adina?
Yes, thank you, Andrei, for your presentation. Well, it was good to have a software engineering perspectives on bias framework. My question would be, do you think that such a framework, which is quite general, would be able to integrate any type of data? For example, you mentioned visual data, and this seems something that is not really very much covered in the existing frameworks. So this is my question.
It's true. Yes, that would be actually a very good thing to succeed in, to integrate non-tabular data, especially images. Now, the ability to integrate that data would actually depend on the availability on two things actually: the availability of algorithms to work with that data, but those definitely exist, and on the ability to actually on the existence of let's say adapters that would be able to that would run the algorithms for images, for instance, in a standardized manner. So standardized or at least uniform formats for data and uniform means of running those algorithms.
Okay, thank you. So now we have some other 10 minutes for discussions and conclusions. So any one of the speakers or of the audience are welcome to step in. So from my point of view, I enjoyed very much all the presentations, and I was just thinking that if we could put in together everything that was presented here, we could just have the base bias approach in AI that somebody would ever think of, because we heard some very different perspectives from both from let's say a more philosophical point of view, but also from a more let's say more experimental point of view with specific cases. And as I understood from the presentation, explainability is also an important tool that can be used in order to identify bias.
Yes, from my side also are covered. It was very insightful for me here all the presenters here, and I get some aspects that I have not really thought a lot of earlier. So thank all the presenters for their contribution and their sharing their insights and their work. So thank you for this.
Yeah, also for me, I would say that it has been so interesting to see, like, you know, so many different also area of research and applications, because sometimes you all know that maybe you do research in a specific field, but also like in the field of bias, there are so many different scenarios, so many different applications, even if we are talking about medical bias. So it has been really, really interesting to see so many groups doing different type of research. And I don't know, I just think it's always great to put together people who are just passionate about something, and, you know, maybe from today's meeting, maybe there will be like future, I don't know, collaboration or research together. So thank you for organizing, you know, this webinar. It has been like a really great opportunity.
Okay, so we thank you, we thank very much all the presenters, all the participants. I thank Sabrina who participated in moderating that. From my point of view, I think it was really, really very rewarding, our participation and in this webinar. We are going to ask all the presenters, if possible, I mean, if they are public, to send their PowerPoint presentation to put on the website of the webinar. And also, as I said in the beginning, the recordings of this seminar will be available on YouTube. And I strongly encourage you to disseminate the recordings of this webinar to your colleagues, to PhD students, because I think that they can really benefit from hearing what it was said today, even if they could not participate in the webinar because of other engagement. So I wish you all a very good day, a very nice weekend, because the weekend is coming, which is a good thing. And thank you again very, very much for your participation.
Thank you, everyone. Thank you, Adina. Have a nice weekend.
Thank you, everyone.
Thank you all. Bye-bye.
Have a nice weekend. Goodbye.
Bye.
This weekend.